but also in the country at large. As mark of its academic excellence, Midnapore College has been conferred with autonomous status by AGC in 2014 and has achieved CP status in two schemes. The specialized status in 2015 is also a vindication to tireless, its tireless and commendable academic strength. The college has been reaccredited by NAC in third cycle in 2017 and has claimed A+ grade with 3.60 CGPA, which is a commendable achievement of this institution. Seminars, workshops, and special lectures, webinars are a regular feature of the college, like that of ours, of which this one day international webinar is a testimony. I extend my sincere thanks to Honorable Research Person for accepting our invitation to join this international webinar of the college. Recent advances in our fight against major human diseases such as cancer, cardiovascular diseases, immunological disorders, etc., owe greatly to the knowledge obtained from basic cell and molecular biology research, which is then used to elucidate the mechanisms of these diseases. By understanding how cells work in the healthy and diseased state, cell biologists can develop new vaccines, more effective medicines, and so increased knowledge may have better understanding of how all living things survive. I hope that participants will be benefited by the enterprising and lucid analysis by our distinguished research persons on their concerned topics to the true spirit of the first area of this webinar, and also by their acumen into their respective fields. I hope the deliberations which will be made will be of great worth for all our participants, and these academic discussions are a bonus to further research activities in this field. Before I request, I would once again like to thank all associated with this academic exercise for taking pains to contribute their valuable deliberations and active participation. I, on behalf of Midnapore College in general and Department of Zoology in particular, thank you all for making this webinar a success. With these few words, I'd like to officially inaugurate this session. Before I end up, I would also cherish that this international exercise will reach the harvest for which it was really intended. Thank you all once again.
Madhumita, your microphone is muted. I mean, could you please?
Okay. Yeah. Thank you so much. Yeah.
You need to tell from the beginning. We didn't hear you.
Yes. Okay. So I would thank Sir for giving out his inaugural address. Now we would be beginning with our scientific session, before going to that, I would again request all of you to kindly keep your microphone muted. This will ensure proper and smooth running of this program. So for the scientific session, we have two lectures by renowned speakers who have extensively worked in the field of cell science. So they will be sharing their experience and guide participants to explore the fascinating world of cell science. The first lecture will be delivered by Dr. Bidesh Mahato, Research Group Leader, Division of Immunology, University of Cambridge, which will encompass very fundamental yet novel and unique findings in the field of cell biology research that will open up future possibilities in cell biology and its application in human welfare. The second lecture will be delivered by Dr. Amitabh Dash, CSIR IICT Hyderabad, who will be talking about the latest discoveries in the applied field of cell research and state-of-art tissue engineering arena with possible applications in near future. I may also announce that following the lecture session, we will open up the floor for questions. So I would request you to post your questions in the chat box, both for this Google Meet room as well as for the YouTube live chat. The questions will be redirected to the speakers by our moderators here. The moderators may also choose some of the participants who are present in the Google meeting room, and they can directly interact with the speakers also. That will be happening after the presentation. Also, there will be a feedback form in the chat box. I would request all the participants to kindly fill up the form and submit it to us by 10 a.m. tomorrow. Okay. You may do it after the presentations are over. So we will be starting now with the first lecture of the day. And before we start with the first lecture, let me give a very brief discussion, introduction about the first speaker, Dr. Bidesh Mahato.
Thank you so much, Madhuchanda, and also thank you so much to Dr. Bera for such a nice introduction. Or did you want
I have, yeah, little more introduction. Can I give it?
Please go ahead. Please go ahead.
Yes, so that the participants will get to know you a little bit more. Yes. Okay, Dr. Mahato has he did his PhD in biochemistry from Indian Institute of Chemical Biology, University of Calcutta. After that, he worked as a research intern in Department of Pharmacology, University in Madison, USA. In the year 2009 to 2011, he was working as postdoctoral research fellow with Wellcome Trust Center for Gene Regulation and Expression, College of Life Sciences, University of Dundee, United Kingdom. Later, he worked as an investigator scientist in MRC, Laboratory of Molecular Biology, Cambridge. From 2013 to 2018, he worked as postdoctoral research fellow with EMBL, European Bioinformatics Institute, and Wellcome Sanger Institute, Cambridge, UK. From 2018 to 19, he worked as a visiting scientist with Wellcome Sanger Institute, and presently he is working as research group leader in Department of Pathology, University of Cambridge, United Kingdom. He has also been awarded with many prestigious awards and fellowship. To name a few, in 2015 to 18, this time period, he had been awarded with CRUK Cancer Immunology Project Award. In 2018, he was awarded first prize for theme poster Cambridge Immunology Forum, Cancer Immunology. In 2019, he was conferred with the University of Manchester Presidential Fellowship. In 2019, he was also awarded Career Development Award by Department of Pathology, University of Cambridge. And in this year, 2020, he has been awarded a project grant from UK RIBB SRC, NSF Bio Lead Agency Opportunity Project Grant, which is in collaboration with researchers from Penn State University. He has published many research articles in many renowned journals, such as Nature Communications, Cell, to name a few. Along with that, he has also published review articles. So I think this is a very brief introduction. We are waiting to hear from Dr. Mahato and his research interest. Let me just tell in brief. His research interest is to study the physiological and pathological role of immune cell-mediated de novo steroidogenesis in regulating inflammation and immunity, and exploit that knowledge for clinical benefit. May we now hear from Dr. Mahato.
Oh, thank you so much, Madhuchanda, for such a nice introduction. And also thanks very much to Dr. Bera for your brief introduction about the college and all those things. So many thanks to Gautam and the organizing committee for choosing me to present my work in this webinar. I am thankful to all of you who are watching this webinar. Thanks for showing interest in my talk. And it's a great honor and a big day for me. And I hope you will enjoy it. So I am aware that many of you are students, so I would like to dedicate first few minutes of my talk for you before I move on to the topic. So let me share my screen with you. Okay. It's very satisfying and I feel proud when students contact me and say that my career path inspires them. Such compliments also inspire me to do something extraordinary so that I can inspire you in the future. Though most of you say that my career path is inspiring, but in reality, the path was actually not smooth or stellar. Rather, it was full with ups and downs, full with failures. But no doubt it was dramatic and I enjoyed it. Starts here. Let me find the pointer. It starts here, and up to the PhD, it was very smooth, straightforward. But to reach here, to progress through this short distance, I had to spend a lot of time. So maybe I wasted my time, but to reach here, I had to travel a lot, mostly eccentrically. And this was simply due to the lack of planning, basically because of not knowing what exactly I had to do to achieve what I wanted to achieve. I was not sure what to do with my life, but I have no regret because I enjoyed the path. A career path in scientific research can be stellar and spectacular if you know what you want to achieve, you have a plan, and you love it. Basically, you have to connect these three dots wisely. Basically, the PhD with the postdoc, and until you be this group leader. So if you can connect these three dots wisely with short period of time, then it's done. Let me put some perspective in the journey. Now you see, I was sailing my ship without a guide, and there was no North Star to follow. So mostly I was just following the coast, and obviously I wanted to enjoy the path. And even if you have well-planned route map, your trajectory may look like this because the path that I have used here was actually a very well-planned journey of Charles Darwin. He actually started from here and traveled the globe following this route. And other things, you know what he had done during his travel. So, and no matter you travel long or not, you just need one solid discovery. Yes, one solid discovery to make your journey memorable. And I wish you all the best, and I'm happy to help you if I can. Okay, let's move into the topic now. To tell you today about why do immune cells produce steroids. The straightforward answer is that I don't know. In fact, no one knows why do immune cells produce steroids. But we are trying to understand why they do so. Over the last 100 years, we know that steroid hormones are anti-inflammatory and immunosuppressive. There is no doubt about it. And the initial works in this field was recognized by the Nobel Prize in Physiology or Medicine in 1950. And it is well known that steroid hormones can control the immune cell function. Now, surprisingly, we never asked whether immune cells by themselves actually produce steroids to control their own function. We never asked such question, possibly because we were blinded by these two bright facts. If the question is about biological source of steroid hormones, we can only think of adrenal gland, gonads, and placenta. And if the question is about immune cell communication or immune cell signaling, then we can only think of cytokine to receptor binding. That means the ligand to its cognate receptor binding, or direct cell contact. So we were unable to think out of the box whether immune cells have the ability to produce steroid or secrete steroid to control their own function. The question came in my mind when I was working at the MRC LMB. You know that famous institute. Every floors and every corridors glitters with several Nobel laureates. So when I was working there, and I was analyzing actually T helper cell transcriptomes, and I found that T helper 2 cells actually upregulate CYP11A1. Not just the mRNA, they also, you can see the protein level of the CYP11A1 is upregulated in Th2, and this is differential upregulation. And in another way, you can see also the proteins by flow cytometry, you can see that the protein is upregulated in T helper 2 cells. So the upregulation of CYP11A1, so CYP11A1 actually is the protein, is the enzyme that catalyzes the first and rate-limiting step of the steroid biosynthesis pathway or steroidogenesis pathway. So it converts cholesterol into pregnenolone, and pregnenolone is the precursor of all other steroids. And by itself is a bioactive steroids, and later we know that it is also immunosuppressive steroids. Even the ectopic expression of CYP11A1 can convert or change a steroidogenic cell type. So the differential upregulation or upregulation of CYP11A1 in T helper 2 cells suggests that possibly those T helper 2 cells are steroidogenic. And that's what we found, both in vitro and also in vivo. In vivo, I mean in worm infection model, we use Nepostrongylus brasiliensis. And on that model, we found that in response to interleukin-4, activated T cells upregulate CYP11A1 and they secrete steroid pregnenolone. And possibly they do so to restore the immune homeostasis. I mean, when worms are cleared, worms are expelled, then to shut down the raised immune response and to restore or resolve the raised immunity, they start producing these immunosuppressive steroids. And here I would like to thank my mentor, Sarah, and my collaborators, Andrew and Vivke. Without their help, it was not possible to do what I did. So the rock-solid discovery was the production of steroid by activated T cells in response to interleukin-4, or you can say that T helper 2 cell mediated steroid biosynthesis. But the claim that they do so to restore immune homeostasis is actually huge. It's a huge claim. And to show direct evidence in vivo model, we had no tools or reagents on that time. Basically, if you want to study in vivo role of the steroid producing cells, you need to stop steroid biosynthesis in T cells or cell type specifically. So ideally, you need a flox mice, I mean the CYP11A1 flox mice, so that you can delete CYP11A1 gene in the T cells. And also, it is ideal to have one reporter line so that you can track those CP11A1 expressing cells or steroidogenic cells in vivo. But unfortunately, these two mice line was unavailable on that time. So I had to take initiatives to create these two mice line. And good news is that both the line are ready to use now. And I am happy to share these two mice line if anybody of you are interested in these mice and interested in immune cell mediated steroid biosynthesis. I also have now generated hypotheses for most of the type 2 immunological context or Th2 immunological context. For example, I believe in helminth infection or in worm infections, these steroidogenic cells are required to restore immune homeostasis. And this steroidogenesis pathway, I mean the immune cell mediated steroid biosynthesis pathway, is dysregulated in allergies and asthma. And that's why you see so much exaggerated immune phenotype because this pathway is mutated or dysregulated. From long time, it is known that pregnancy is a type 2 immune phenomena, but it is not known exactly what are the contributions from those T helper 2 cells. I believe that those T helper 2 cells or other steroidogenic immune cells actually promote pregnancy by supplying the precursor of progesterone. But all these are hypothetical and need experimental evidences. And this immune cell mediated steroid biosynthesis may be relevant to wound healing, mucosal tissue homeostasis, atherosclerosis, arthritis, parasitic infection, and maybe the model you study. For example, I'm lucky that Professor Richard Grensis is happy to help me with the trichuris infection. As you can see, the infected mice, I mean the trichuris infected mice actually develop these steroidogenic immune cells, but and those are absent in the control mice, but we don't know who they are and what they do. Not just immune response or immunity, these steroidogenic immune cells may have role in normal hematopoiesis or maintaining normal immunological homeostasis. For example, you can see this CP11A1 expressing cell in the bone marrow, and they are basically the mast cell progenitor, basophil progenitor, and lymphoid progenitors. But again, we don't know its role in normal hematopoiesis. So I believe that this discovery of immune cell mediated steroid biosynthesis is a landmark discovery. No doubt this is a breakthrough in cell science, but we need more and more evidence supporting that claim. So no matter what context we are studying, the core strategy would be very simple. Use the CP11A1 mCherry mice to track those steroidogenic immune cells, identify them and characterize them, and then use CP11A1 flox mice to test their biological role, because you can delete CP11A1 cell type specifically and you can check their role. So the core strategy to study immune cell steroidogenesis is going to be very simple. And rest of my talk, I will focus on anti-tumor immunity and cancer immunotherapy, because it was 2014 when we published our first report. And in 2013, the cancer immunotherapy was awarded as the breakthrough of the year 2013. So I could not resist myself to test the role of these steroidogenic immune cell in the context of cancer, how they control anti-tumor immunity. So we know that we have anti-tumor immunity, and our immune cells have the ability to identify and kill those cancer cells. And they do so very efficiently, but not always. Sometimes they fail. And when they fail, the tumor becomes established. And in established tumor, it is unfortunate, but this is very true, that the immune cell promotes the disease by creating that microenvironment more and more immunosuppressive. Now, if we know those immunosuppressive mechanisms, then it would be easier to develop cancer immunotherapy. And the fact that were very well known at that point is that T cells are energetic and dysfunctional when tumor is established. Dendritic cells are tolerogenic. Macrophages show type 2 phenotypes. Those actually help in further immunosuppression. Those were known actually. And immunoregulatory number of the regulatory T cells increased on those established tumors that are actually immunosuppressive. Now, another fact was very clear, that many tumors actually show type 2 phenotype, type 2 immune response. So Th2 cells are there, interleukin-4 is there. So I thought maybe steroids are there, because Th2 cells actually produce steroids in response to interleukin-4. And you know that steroids are immunosuppressive in general. And when I was going through literature, I was surprised that steroids actually can make T cell energetic and dysfunctional. They induce type 2 phenotype in macrophage, and those macrophages are called actually M2C macrophage. And in presence of steroids, dendritic cells are actually tolerogenic dendritic cells. And it was also known that in presence of steroids, the number of regulatory cells can be increased. So all these facts were known from other immunological contexts, not from the tumor, but there are many published reports that steroids actually increase the regulatory number of the regulatory T cells. They induce tolerance in dendritic cells. They induce type 2 phenotype in macrophages, and they make T cell dysfunctional. So I was very excited to hypothesize that immune cell mediated steroid biosynthesis in the tumor microenvironment suppress anti-tumor immunity and promotes the disease. And when I'm testing this hypothesis, I was interested to study the upstream signaling cues that induce steroid biosynthesis, and also how they suppress anti-tumor immunity. The effect of those secreted steroids in immune cell function. And if we know this pathway well in the context of tumor, then we can find or identify the targets for cancer immunotherapy. It can be directly CP11A1, it can be upstream pathway, or it can be downstream. Thanks to Jackie and Lucy, who helped with their in vivo models. And we used here B16F10 melanoma model. We choose melanoma because in human, it was known that melanoma actually induce type 2 immune response, and it also induce type 2 immune response in mice. And to model the primary tumor, we actually inject B16F10 cells subcutaneously. And to model the lung colonization, the lung metastatic dissemination of cancer cells, we inject the B16F10 in the tail vein. And when we analyze the subcutaneous tumor, the subcutaneous B16F10 tumor and associated immune tissues, you can see that actually tumor induced the CP11A1 expression in T cells, both in CD8 positive T cells and also in CD4 positive T cells. So these are actually possibly T helper 2 cells, and these are TC2 cells. But surprisingly, we don't see the expression of CP11A1 in the lymph nodes or spleen. This is the brachial lymph node, was the tumor draining lymph node. So those steroidogenic immune cells are restricted or confined within the tumor. So in next, we wanted to check whether there are any other immune cells that are steroidogenic, because this experiment was done by flow cytometry. So in next, we wanted to find an unbiased or global view of the mCherry or CYP11A1 expressing cells. So we decided to do single cell RNA sequencing. So what we did, we actually inoculated B16F10 tumor in the CYP11A1 mCherry mice. And as you can see that there are many cells that actually upregulate CYP11A1, and you can detect it from the mCherry signal. So we purified those cells into 96 well plate, 79 from the positive, I mean the CYP11A1 mCherry positive, and 15 from the negative. These negative cells were just for the analysis purpose, and we had 2 well for technical control. Now, and then we did SmartSeq2 single cell RNA sequencing, and this is the cluster annotation. These are the basophil eosinophils. These are the myeloid cells, mostly the macrophages. These are the mast cells. And these are the 3 population of T helper 2 cells. This is revealed by the cluster annotation, but they are basically the one big cluster of T helper 2 cells. Only the cluster annotation reveal them as a 3 separate cluster only because few cytokine genes were differentially regulated. But that can happen in T helper 2 cells. Some cells express more interleukin 13, some cells express more interleukin 4. So I consider all these 3 clusters as one big T helper 2 cluster. And this TC2 is actually CD8 positive, but GATA3 expressing cells. So I call them TC2. And these are the B16 cells. Now, if you look into the mRNA expression, I mean, into the CYP11A1 mRNA expression, the most CYP11A1 positive cells are basically T cells, that are the TH2 cells and TC2 cells. Though we also observe that the basophils and mast cells have the ability to upregulate the CYP11A1. But in case of tumor, mostly they are T cells. And you can see the protein expression, they are mostly the T cells. So in next, we wanted to delete this CYP11A1 T cell specifically. And to do so, we used CD4-CRE. And you know that when T cell develop in the thymus, it has one double positive state that express CD4 and also CD8. So that means even if you use CD4-CRE, it actually deletes CYP11A1 both in CD8 positive T cells and CD4 positive T cells. So all T cells are actually deficient of this CYP11A1 and they don't produce steroid. Now let's see what happens in tumor growth. And we found that if you stop steroid biosynthesis in T cells, by deleting CYP11A1, the tumor growth is restricted compared to the control group. And this was significant. So, and if you add back pregnenolone, for example, this is the normal tumor growth in control mice. Now, this is the tumor growth in the knockout. Knockout means the T cells are deficient in CYP11A1, so they are not producing steroids. So this actually restricts the tumor growth. Now, if you add back pregnenolone, then the tumor volume actually increase and comparable with the control, this and this. So you can reverse this tumor restriction phenotype by applying pregnenolone into the tumor site, I mean topically. So that means pregnenolone actually complements CYP11A1 deficiency. That means the effect that we see, the phenotype that we see was because of CYP11A1 mediated biosynthesis.
Yeah. Sorry to interrupt you with this. You have to present your screen again. Somehow that has been escaped, right?
Okay. I mean, can you see now? Hello,
Not yet.
Gautam?
No, not yet.
Can you see me?
Not yet.
Not yet. Not. We can't see.
Can you see? Hello, Gautam?
Yes, it is.
Yes.
Yes,
yes.
yes,
Yes,
it is. It is visible now. It is visible now.
Okay. So
Okay, please
can
continue.
you see this slide now? So sorry for this interruption. I don't know what happened.
No, that is due to some technical glitches. Someone else has started presenting, right?
Okay.
Okay.
So
Carry on.
okay. So what we saw that if we stop steroid biosynthesis in T cells, then actually tumor growth is restricted, and that was quite exciting. So we wanted to know basically whether that effect is coming from the steroids or just other function of the CYP11A1. So we wanted to just complement by pregnenolone. If we can complement that CYP11A1 deficiency by pregnenolone, that means that the phenotype was because of steroid biosynthesis. Because we are not sure CYP11A1 have any other, I mean, has any other role or not. So as you can see, when we stop steroid biosynthesis in T cells, tumor volume was significantly reduced with the control. And then when we add back pregnenolone, it was restored. So that means it was because of the steroid biosynthesis. And similar phenotype we observed in the lung colonization model. I mean, in the model where we observed metastatic dissemination of the cancer lung. As you can see, when we deleted CYP11A1 in the T cells, then the metastatic spots were significantly less than the wild type control or the CD4 control. Okay, let me. And to explain these two phenotypes, so far what I know is that this is because of regulated entity. I mean, when we delete CYP11A, basically that stimulate or boost anti-tumor immunity. So as you can see, for macrophage, this is the control mice. In control mice, the tumor actually 2 macrophages are more. They are type 2, I say, because they express arginase 1. And number of M1 macrophage is less because they express INOS. So number of M1 macrophage is less, and M2 are more. And these M2 macrophage actually promotes tumor growth by suppressing anti-tumor immunity. Now, if we delete CYP11A1 and stop steroid biosynthesis in T cell, then these good guys actually increased in number. I mean, the number of M1 macrophage increased, but the number of M2 macrophage, these are the bad guys, their number is decreased. So that indicates a clear upregulation of anti-tumor immunity. The similar thing happens in CD8 positive T cells. Those are crucial for anti-tumor immunity. Actually, you know that cytotoxic T lymphocytes are very important in tumor. So you can see when we deleted CYP11A1, then actually they upregulated more, I mean, more interferon gamma expression. And we also checked other properties like degranulation of the T cells. You know that when T cell kill cancer cell, they actually bind with them and they degranulate their perforin and all other stuffs, and they destroy the cancer cells. So we wanted to check whether they are degranulating more in the knockout mice or not. And we found that those degranulating T cells are more in knockout mice. And we checked this based on the CD107A expression. This is a known marker of T cell degranulation. So it seems that deletion of CYP11A1 in T cells actually stimulating anti-tumor immunity. And we also observed that number of regulatory T cells are less in the tumor of knockout mice. That's also good. So there are clear indication of boosting anti-tumor immunity. So if I summarize all those findings, that first I told you that in control, M1 macrophages are more, M2 macrophages are more, but in knockout mice, M1 macrophages are more, and the T cells are mostly dysfunctional in the control mice. But if we delete and stop steroid biosynthesis in T cells, then they are more functional. They are producing interferon gamma and they are degranulating. I haven't shown the TNF alpha, but we also observed that they are secreting more TNF alpha. Number of regulatory T cells are more in the control mice in normal tumor, but the number of regulatory T cells are less. And also we observed that natural killer cells are more functional and degranulating more in the knockout mice, but I have not shown the data. And bioinformatics prediction tells that in human, many tumors are actually steroidogenic based on CYP11A1 and other gene expression. Not in all patients, and not in all tumor types. Patients in nearly many tumor types actually upregulate steroid biosynthesis pathway. But that's hypothetical based on the bioinformatic analysis. So the next step would be to develop and establish a diagnostic platform to stratify the steroidogenic tumors so that we can stratify patients and to develop a treatment strategy to inhibit the intratumoral steroid biosynthesis pathway. And good news is that drugs are available to stop the steroid biosynthesis, and that can be used pharmacologically, but needs further research. So if I summarize so far, whatever I have told you, the big thing is that activated T cells actually can produce steroids in response to interleukin-4, and that is in the context of tumor and anti-tumor immunity. Tools and reagents are ready. I mean, these two great animal models are ready to study immune cell mediated steroid biosynthesis, and I'm happy to share mice if you are interested in this immune cell steroid synthesis. I showed you that deletion of CYP11A1 in T cell restricts tumor growth and also the metastasis, and this is because of stimulation of the anti-tumor immunity. It actually increase the functional T cell, increase the number of functional M1 macrophages, and decrease the regulatory T cells. And highly likely it is true in many tumor types. Highly likely it can benefit many patients, but needs further study. So before I move next, I would like to tell you that there is no reason to think that immune cell mediated steroid biosynthesis is specific to T helper 2. It sounds that it is specific to T helper 2 because I have done some research in T helper 2. But there are possibilities that maybe mast cell, basophil, B cells, and monocytes may have the ability to upregulate CYP11A1 and produce steroids in different immunological. We don't know about their role because we have not tested. So please look into the CYP11A1 expression in your immunological context that you study. I'll be very happy to help you solve some of your questions maybe. And before I stop, I would like to thank my mentor again, Sarah Techman, and my collaborators in Vivke, Jackie, David, and Klaus, who actually believe in this work. And I'm lucky that I got their help. And I'm thankful to my colleagues who helped me doing experiments. For example, Juma, Lucy, NG, and very instrumental publishing all those reports. And Richard, Ziwe, Mingfu, and Gojde are my new collaborators, and we are excited to explore this new field as a team, as a big team. And before I stop, let me introduce my brand new team, pioneering immune cell steroidogenesis. So I'm lucky that I got Megan and Christopher, two very bright and brilliant students. And soon, within October, I'm going to recruit another very curious postdoc. And I'm looking forward to fill my lab with observers, thinkers, doers, and obviously the daredevil risk takers. And my lab is funded by National Science Foundation and BBSRC and University of Cambridge at the moment, but I am hoping to attract more and more funds to support my research. And thank you for listening, and I will be happy to suggestion, advice, and answer the question you may have. Thank you.
Thank you, Dr. Mahato, for your nice lecture. We may now take some questions. I would request to kindly keep your microphone muted. I would request to keep your microphone muted. If you have any questions, please write your name and affiliation in the chat box so that we may connect you with the speaker. But please kindly keep your microphone muted. It is creating problem. I will now hand over this session to the moderators who would be asking questions. Can you please pass the questions?
Hi, am I audible?
Yeah.
Okay, so there are so many questions. One from two platforms, one in this room, and another from the YouTube session, YouTube part. So I am first putting the questions from here. Dr. Supriti Sarkar, she has made a question that it is visible in the chat box. Do immune cells produce steroids in case of any type of tumor, or it can do in case of other types of infections too? Because can you please have a look in the chat box? The questions is typed there.
Okay, so Supriti Sarkar.
Yeah, Supriti Sarkar. Right.
Supriti Sarkar.
Yes.
Oh, do immune cells produce steroids in case of any types of tumor, or can it do in case of other type of infection too?
Okay.
Yes. Answer is yes. We checked melanoma model and also checked breast cancer model. Supriti, can you hear me? Or
Very
can
much. Okay. Yeah. I can hear you.
Okay. So answer is yes. We checked melanoma model and also breast cancer model in mice. They actually produce steroid, but
I am interrupting you again. Videsh,
yeah, sure. Yeah,
kindly just turn off your PowerPoint presentation. Otherwise,
okay.
You are not visible, right? Either leave presentation.
Okay.
Yes.
It's fine now.
Yeah.
Now it is okay.
Yes, fine.
Yes, it is okay. Yes. Yeah.
So we checked breast cancer and melanoma in mice, but we have not checked human yet. Not checked means we checked it bioinformatically, and there are indications, but we have not done any experiments using human cells or something like that. But as immune cell steroid synthesis is a two immune phenomena, So you can expect in any type of immunological context, particularly in not just in tumor, but also in type 2 immunological context that are actually upregulate or induce T helper T cells, mast cells, and basophils. I think this is what you wanted to know.
Yeah, thank you.
Okay, the next question I'm putting before Dr. Bidesh Mahato is that it's from Dr. Orunima Datto. She is from SGGUCG. I'm not getting the getting actually. So the question is, can CYP11A1 mediated suppression be equally effective for estrogen mediated cancers?
Yeah, I saw that question. Yeah, I see that question now. Yeah, that can be disaster. I don't want to think of that possibility, Orunima, but it can be. It can be in breast cancer. It may be the case that the tumor is converting that pregnenolone into the estrogen, and if that is the case, then it can be disaster because the tumor respond to estrogen. It can drive the tumor progression and metastasis. But obviously there is a possibility, and for example, in prostate cancer, what happens even if after castration, when the androgens are not available, still those cancer actually turn into a androgen the castration resistant tumor. And what are the source of the steroid hormone is not known. There are few reports telling that the prostate cancer by themselves producing steroids, but maybe the immune cell have some role there, and not just the immune cell mediated steroid biosynthesis. With the course of the tumor evolution, actually maybe those immune cells also inducing steroid biosynthesis in the tumor cell. This can happen, but all are actually hypothetical. So the answer is yes. It may be the case, but we need further experiment to check it whether actually the estrogen, we are producing estrogen or not.
Okay, Dr. Bidesh Mahato. So next question is from Barsha Das Sharma. Can you please say it in the chat box?
Oh yeah. So
okay. She has not put any question. Just a minute. Let me check. Yeah, Dr. Acharya Ardha. He has made a long question. Can you please read it out and answer?
Oh yeah, sure. So let me read it out. Hi, I am a student and I would like to put forward a doubt to you, sir. CYP11A1 also encode a member of the cytochrome P450 superfamily of enzymes. Not also actually Acharya, CYP11A1 actually encode the P450 SCC monoxygenase, yeah, which catalyzes many reactions. No, that actually is not the CYP11A1. CYP11A1 express only P450 side chain cleavage enzyme, and the monooxygenases are actually synthesized by other gene. I think you are telling about CYP19. So CYP19 actually encodes monooxygenase, and that actually catalyzes others. So lipids. So how can we address the adverse effect of deletion of CYP11A1? Yeah, so, but it can be a very good question. CYP11A1 actually not encodes monooxygenases, but monooxygenases can convert some other steroids actually. Now, when you inhibit CYP11A1, it also actually inhibits those monooxygenases. So if you inhibit, for example, CYP11A1 by aminoglutathione, then it also inhibits CYP19 or monooxygenases. So to find a specific inhibitor of CYP11A1 is difficult, but it is possible to find. And remember, the steroid biosynthesis rate in immune cell is very low compared to the adrenal gland or gonads. So a low dose of the inhibitor can successfully inhibit CYP11A1 compared to the other steroidogenic cells. And now the main aim is to stop steroid biosynthesis. So if we study immune cell mediated steroid biosynthesis, highly likely we can find differentially regulated pathway. So we can target those differentially regulated proteins, and we can check it specifically in immune cells. So, but yes, you are right, by using aminoglutathione or the inhibitor that also inhibits CYP11A1 and other monooxygenases is difficult and may have side effects.
Okay, Dr. Bidesh Mahato, the next question is from Kaushik there. He is an student of our department. The question was a bit simple one. Is there any age factor related to the expression of CYP11 gene?
The straightforward answer is no, we don't know. So, but maybe because steroid hormone biosynthesis affected with old stage, the thymic function is also affected with older stage. So there might be effect of age, and it's a very important question because cancer mostly an old age disease. So we need to know whether it has any effect with the aging. So thank you so much for pointing that question.
Okay, so thank you, Vidisda. And the next question is from Dr. Shubho Ghosh. He is a faculty for department. As all steroid hormones are synthesized in a common biosynthetic pathway, are there any possibilities that the steroid suppressing drugs can hamper regular endocrine endocrine functions?
Oh, it's a great question. It's a great question. Yeah. So if you stop steroid biosynthesis non-specifically, then you may have deleterious effect by suppressing the steroid biosynthesis in the adrenal gland or steroid biosynthesis in other tissues. So ideally, we need to find a specific way to target this pathway. So that can be possible if we study the differentially regulated pathway. And for cancer, we can do in a different way. So you know that adoptive cell therapy is showing magic in the clinic. For example, the T cells, adoptive transfer of T cells, and also adoptive transfer of dendritic cell showing great promise against tumor. So now if you think from the clinical point of view, if tumor is full with steroids, then those T cells or dendritic cell will not work because of the steroids. Steroids will suppress them. I mean, they will make T cell dysfunctional and induce tolerance in dendritic cell. Now you can make those cells steroid resistant because it is not just the secretion of the steroid, but it also the response from the immune cells. So if you can make those immune cells steroid resistant, then those can make those can work like magic bullet. So now if we combine together, I mean combine the two things, so we are reducing the steroid level by using drugs or inhibitors of the pathway, and we are using the T cell or dendritic cells for the therapy that are resistant to the steroid. So combining effect can bring a breakthrough success against tumor. But it's true, all are hypothetical. We need more experimental evidence.
Okay, thank you, Vidisda. The next question is from Samarpan Maiti. He happened to be our student now doing research in IICB. the question is, medicines which have steroidal properties can induce cancer propagation, as there may be regular medicines which are actually different kinds of steroids.
It's a great question, Samarpan. I mean, in the clinics, doctors actually use steroids to treat the side effect of the cancer patients or cancer treatment. So what happens? This is basically to manage the disease, to reduce the inflammation or the pain that patients are having. So then they prescribe steroids to reduce those pain and exaggerated immune phenotype. So now, not very recently, during the last few years, actually there are also built up evidence that if you use steroids as a disease management, then the immunotherapy actually don't work properly. But doctors, good thing is that doctors use that temporarily, so you are not using steroids for longer time. So we have to think alternative. I mean, but this is very true that use of steroid have detrimental effect if the patients are having immunotherapies. So maybe non-steroidal, and even in the immunotherapies, doctor actually prescribe steroid because immunotherapy sometimes elicit hyperactive immune response, and to check that and to control that, doctor actually prescribes steroids. So to check immune response, temporary use of steroids, I think okay, but all those things need drug trial. And thank you.
Okay, thank you, Bidesda. I'll ask Dr. Gautam Ghosh, how much time do we have? Because I feel that Bidesda is getting bit of tired by so much of questions.
I'm not tired. I'm excited. I love my audience.
Oh, we can go for another 2-3 minutes.
Another 2 minutes. Okay, I'm now putting the questions from YouTube platform because there are so much of questions. So I would like to put some of them in the chat box.
Another question.
Yes.
I have a question for you.
Okay.
That I am posting in chat box.
Okay. Dr. Gautam has asked, can you please read it out, Gautam, Bidesda?
The last one.
So the last one.
Yes.
Yeah,
So that's from our Dr. Gautam Ghosh. There are huge array of signaling molecule produced by immune cells. Yes, that's true. Almost to execute almost all type of communication function. Yes, that's also true. In such scenario, what are the selective advantage of T cells to produce steroids like a small group of odd men out?
I just failed to type one word. So almost the huge array is for the peptides, right? So almost all of the signaling molecules through which the immune cell interacts are through peptides, right? So what is the selective benefit or advantage of synthesizing steroids?
Yeah.
And that can be harvested by the T cells, right?
That's one of the most important fundamental question of immunology, Gautam. That one of the most fundamental question in immunology, why they do so. Not just one cytokines, they have enormous ability to secrete many cytokines and at any given time. But what they are doing and when they are secreting what and why, these are actually big questions in the field. So I just want to focus my research only on steroids, when they actually start secreting steroids and why they do so, we don't know. So I want to test this in vivo worm infection model or in real immune challenge model. For example, trichuris infection or Nephrostorongialis infection, and look into the T helper cells, whether the normal T helper cells or they are different type of T helper cells. why they are actually upregulating CYP11A1 and producing steroid, and what the physiological role. I am very much sure T cells or any cell is not producing steroid to feed a tumor and to grow it. I mean, it's a maladaptation. The physiological role must be different. So in T helper 2 cell, it can be to restore immune homeostasis. So those immune cells actually what to say? I mean, getting signal that once are cleared, so that they are producing steroid to restore immune homeostasis. Maybe they are secreting steroid to resolve the other innate immune cells, or maybe to wound healing, the worm or any other infection make tissue damage. So maybe they are secreting steroid to repair the damaged tissue as a wound healing process, but all are basically hypothetical. I don't know why they are secreting steroid. In cancer, it may have huge implications. We can save a lot of lives, so that's why I prioritize to study its role in cancer immunity, and that's also at the moment major focus. But at the same time, I am developing collaboration to answer your question with many other groups like Professor Richard Bensis and many others to look into its real or physiological role, not the pathological like cancer. So thank you so much for raising that. It's a very fundamental question.
Okay, some other questions now I'm taking from YouTube platform. So the first question is from Abronil Gangopadhyay, who is an MSc student in virology, National Institute of Virology. What fluorescent dyes are used to detect cells in flow cytometry? Videsda, you can read it in the chat box. It is from it has been posted by me, but it is from Abronil Gangopadhyay. What fluorescent dyes are used to detect cells in flow cytometry?
So I don't see it, but let me tell you. So is this to detect CYP11A1 or any other protein?
You can't get the answer directly from there because the question is from YouTube platform, so you can't get it directly from them. So you have to answer from this part on.
Okay, if the question is to detect CYP11A1, then we use actually CYP11A1 M-cherry mice. So M-cherry actually fluoresce, and that we detect in the flow cytometry. And we also use another antibody. Unfortunately, you cannot buy a conjugated antibody for CYP11A1 because that's not available. But you can tag fluorescent or conjugate fluorescent dye to the CYP11A1 antibody. But there is no primary antibody available conjugated with any fluorescent that mouse line. And if your question is to detect any other proteins, then specifically I cannot tell you now because we use several fluorescent dye to detect several surface protein and also many other things.
Okay, the next question, as you can see in the chat box, is from Kumaresh Mandal, who is from University of Calcutta, but I don't know whether he is a professor or a student. Can CYP11A1 restrict lung metastatic dissemination? From Kumaresh Mandal, you can see that.
Can you read that out for me once again?
Yeah, the question is how can CYP11A1 restrict lung metastatic dissemination?
Oh, that's more than 1 million pound grant. I wrote that down. Yeah, that we don't know. It may be just simply because of immune response, but for the lung metastasis, it may have a role in direct dissemination of the cancer cell into the lung. So, equal opportunity, I don't know. but I have to test it because I have the mice models and reagents ready, so I can test it. But it's a great question, and also I wrote grant to analyze that how actually immune cell mediated steroid biosynthesis promoting cancer metastasis. If you can look into the recent Nature paper, you can find a report in Nature that tells that glucocorticoids promote breast cancer metastasis. Glucocorticoids promote breast cancer metastasis, and they show that actually upregulation and an elevation of glucocorticoid levels actually promotes the metastatic dissemination of the breast cancer cell into the lung. So, but they have not shown the source of those cells. I believe on that system also the immune cells, some of the immune cells, immune cell mediated steroid biosynthesis have role. I mean, the source of that glucocorticoids may be coming from the immune cell mediated steroid biosynthesis. And how they do so, the exact mechanism, the detailed mechanism is not known, particularly from the point of view how the steroidogenic immune cells contributing. So I will surely test that by next couple of years. Thank you for raising that question as well.
Okay, the next question is from Mr. Anup Kumar Das, but I didn't get his affiliation. Is there any other model? Rather than these CD4 CRS and CYP11A1 for this study? Is that clear?
Is that in the chat box, that question?
Yes, that question is from Anup Kumar Das.
Anup Kumar Das, I'm sorry.
Oh, so much excitement. Actually, I cannot find it now. If chat box is full with question, I'm so happy.
Let me read out again.
Yeah.
Is there any other model for studying these CD4 CRS and CYP11A1 study?
Yes, you can study the immune cell mediated steroid biosynthesis in any other model. I think you wanted to. I think he or she.
It's he. Anup Kumar Das. It's he.
Okay, so I think he wanted to know whether we can delete CYP11A1 in other cell type. Yes, flox mice is available. CYP11A1 flox mice is available. So you can cross it with any other key driver, and you can delete it in other cell types. So, but you have to look into the literature and finalize what you want to do with these models. But yes, it is. These two models actually facilitating your research, but this is not confined to this mice model that I use. You can use any mice model. You can use human patient sample of any immunological context. I mean, or cell types or cell samples, but only these basically facilitating the research, not the only way to do immune cell steroidogenesis. I will encourage you, whatever model is available to you, please look into the immune cell mediated steroid biosynthesis, particularly CYP11A1.
Okay, the next question is from. Next question is I have just pasted in the chat box the last one. It will be easy for you to get it.
Okay.
If, I wanted to know if you have observed whether CYP11 blocking induces immunotherapy susceptibility in tumors.
Okay. Parthoputim Jan, you have observed.
Okay, this is not my question. I have actually got it from the YouTube platform, and I have pasted it in the chat box. Okay. This is
who is that?
This is from Mr. Tomal Sarkar, who is an MS student in NIBMG, Sollani.
Thank you, Tomal, for asking this. So your question is that if you have observed whether CP11 blocking induces immunotherapy susceptibility in tumors.
Right.
Okay, that's a great question actually. No, I have not tested it, but I will do so. I will conduct that experiment very soon. So to conduct that experiment, you need the ethical approval, and I'm on the process. And I'm collaborating with other people to check whether it can be combined with the anti-PD-1 particularly. I think yes, because if you know that, I think you know that B16F10 actually doesn't respond to the immunotherapy well. So, but it's responding to the CP11A1 inhibition. That means we can combine the immunotherapy. And now I think you wanted to know the immunotherapy with the checkpoint blockers, but there are many other immunotherapies I told you that can be integrated. For example, you can make your dendritic cells steroid resistant. So basically, you are making them steroid resistant so they can respond well. And also you can make your T cells steroid resistant. So those are also immunotherapy. So highly likely it can be integrated with other immunotherapies. But we have to remember that all these things need actually preclinical trial and then in human trials. So, but I'm very much excited to see those results actually on those models. Thank you for raising that point as well.
So we have to conclude now, right? One last question I am asking on behalf of Khushbu Chaturvedi. Obese people, you can understand why I am interested in this. Obese people have higher steroidogenesis and have weaker immunity than normal people. Does this make them more prone to cancer as well? If yes, does this affect their recovery time from the disease?
The question is in the chat box. The question is in the chat box, the last one.
Okay. So, from the Khushbu.
Yes, from Khushbu. Yes.
Obese people have higher steroidogenesis and have weaker immunity than normal people. Does this make them more prone to cancer as well? Yes, does this affect the recovery time from the disease? Maybe or maybe not. We don't know. We don't know, Khushbu. This is good hypothesis. So someone need to test that. I mean, it would be good hypothesis. Yes, so it is known that in obese people M2 macrophages are active, and I'm not a scientist of that area. My knowledge is very poor. But if so, then yes, it may have some role. But someone need to test that. Sorry that I could not answer exactly because I don't have experimental evidences. But that's a good question. Someone need to test that. Yeah.
Okay, thank you, Gautamda. Thank you, Videsda. There are so many questions you have answered, and I think there's too much of time we have taken from you. So over to Dr. Madhuchanda Banerjee.
Well, thank you so much. I mean, it was a very interesting session, and there were so many questions. Questions are still many more. They will remain, but it was a very lengthy session for you also. So thank you so much.
Now I love my audience. I love questions. So I will try to answer. I will look into the YouTube and try to answer my questions. And it was a great experience spending time with you. And it was a big day for me. Thank you so much for inviting me.
You just forgot one thing.
What?
You just forgot to mention one thing. That you are student of this college.
But I mentioned that. I showed in the career path. I was student in the Midnapore College.
That makes it even more special.
Yeah.
Thank you so much. Thank you. We are delighted to have you here. Thank you so much. So we will now proceed with the scientific session to the second lecture. And so the second lecture will be delivered by Dr. Amitabh Das from CSIR, Indian Institute of Chemical Technology. I would request my colleague, Dr. Amitabh, to kindly introduce him.
Good afternoon, everyone, and thank you very much for being us here today. Thank you, Dr. Mahato, for an enlightening presentation. I am Momita Adok, Assistant Professor of Midnapore College, Department of Zoology. It's an honor for me to introduce our next speaker, Dr. Amitabh Das, Principal Scientist at CSIR, Indian Institute of Chemical Technology, and Associate Professor with the Academy of Scientific and Innovative Research, India. Dr. Das completed his doctoral research from CSIR, Central Drug Research Institute, India, following which he did his postdoctoral training from Baylor College of Medicine and Mayo Clinic, respectively, USA. He was an assistant professor in Regenerative Medicine Division, Department of Medicine, and held joint faculty position at Biochemistry Division, Department of Basic Science, Loma Linda University, USA, before returning back to India in 2011. Presently, the research focus of his lab is basic stem cell biology and regenerative medicine using stem cell gene therapy approach for tissue regeneration and development of novel translational therapeutics against cancer relapse or recurrence and liver disease. He has published more than 45 research papers in reputed peer-reviewed international scientific journal and 1 book chapter. He has 1 US patent and 2 Indian patents in his name. He has been serving as an assistant editor of International Journal of Surgery. 5 research scholars have been completed PhD under his supervision. So without taking much time, I will hand it over to Dr. Das. Dr. Das?
Yes, very good afternoon to all of you. At
Hello,
our
Das.
outset, let me thank. Yes, I'm already there, but I'm not able to share my screen. Can I just come back? Am I audible?
Yes, you are.
Am I audible?
Yeah.
Yes, yes.
Okay, I'm not able to share the screen. It just happened once, but in the past too. Let me just see. Not able to. Yeah. Is the screen visible?
Yes.
Hello?
Yes.
Yes, sir, it's visible.
The screen is visible?
Yes.
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 Okay,
Yes.
wonderful.
Yes.
Yes, visible.
Yes.
Okay, at the outset, let me thank, of course, my son who just now helped me. That's the generation actually belongs to, and very thanks to the organizing committee for inviting me to deliver this webinar and share the recent happenings going on in my lab. Before I start with, let me thank your faculty, Dr. Kajol Mojumdar, who happened to be acquaintance known to my wife, Dr. Chandra Dev, and through them, they contacted me to participate in this particular webinar and deliver the lecture. So Thanks for introducing me, Dr. Amitabh, and also to begin with, as I say, my lab interests are mostly governed with stem cell basic biology and tissue engineering, and that takes into different types of tissue specific cells that we work on for developing regenerative therapies. Mostly we use induction models or the stem cell gene therapy models to work on. As has been introduced, my lab also looks on the molecular signature of the cancer stem cells that participate in the EMT, metastasis, and various other phenomena using mostly the lab is not only looks into the in vitro cell culture models or the molecular mechanisms, but also we use mostly preclinical models for every disease or every study that we work out. We use preclinical models to generate the proof of concept, and of course we are already into collaboration with the various clinical partners to make our developed therapies to see the light of the day. So today, in today's lecture, already we had a wonderful session earlier. Our presenter was presented a wonderful work, and was very much delighted to hear him. But my topic, as I work in the Ministry of Science and Technology, Government of India, under the CSIR chapter, we are more governed. We are more into developing translational therapeutics. We are more into developing the translational biology. So that the basic biology, what we work on, we take it to the part where the translational aspects are being worked out, so that can be help for the development of drug therapies or therapeutics. So my today's talk, I will mostly talk about the stem cell basic biology and the tissue engineering portion for regenerative therapies. So before we begin with the present situation in recent advances being made in the induced pluripotent stem cells, where we know that Shinyaka and his co-workers were awarded the Nobel Prize for these particular development. But nevertheless, the adult stem cells, which have been discovered for a long period of time, for been already there in the clinic, been tried in the clinic for several different types of therapies. One of the most important example about it is the bone marrow derived bone marrow transplantation, which is nothing but other than it's again stem cell transplantation therapies that has been developed. But why do we study this adult stem cells? Because adult stem cells are present in the body, and in adult body, in every tissue harbors some or the other kinds of other adult stem cells, and that stem cells has the potential to replace the damaged cells that has been occurred due to the illness, and that can be utilized to generate effective translational therapies. So adult stem cells, although I say it's present in every tissues of the organ of the body, but it is very, very difficult to identify, isolate, purify, and the benefit of these is that, on the contrary, the benefit of these as compared to the embryonic stem cells is that these cells do not have any ethical issues attached to it. So the utilization of these adult stem cells can be very faster once one person identifies and isolates and purifies these particular stem cells for developing clinical therapies. But there are, as like every studies, there are the thumb rules for these kind stem cell biology, where we first of all do an isolation of these cells. These isolated stem cells can be needs to be characterized well. Once the characterized cells are there, it should have the expansion capacity in the in vitro or in vitro culture systems, for that they can be utilized for transplantations. Second, the last but not the least about this, all these techniques is the differentiation. That means any adult stem cells that are isolated or purified and characterized, they need to differentiate in the tissue-specific lineages. That is the most important part. So how do we study these particular 4 different steps in the stem cell biology? Isolations of these stem cells can be done from the tissues. Every tissue, adult stem cells have expressed out CD markers like CD20, CD29, CD44, CD90, CD118. There are several different CD markers that are expressed on the cell surface of these stem cells, and these markers can be utilized for harvesting, for isolating these cells by using either the flow cytometric FACS sorting systems or the magnetic activated cell sorting systems. So in my lab, what we generally do, we identify as like any other stem cell biologist, what we do is that we try to identify different markers, because so far till date in the literature, you will find there are various different markers that have been used to identify or isolate these stem cells. But these stem cells do show the properties of differentiation capacities. That is one of the proof of principle for utilizing calling them as stem cells. But there has been no such consensus systems other than very few markers which has been categorized that these are the one which have been the markers of the say mesenchymal stem cells or hematopoietic stem cells or any other tissue specific stem cells. So in my talk, I'll be initiating out with various different types of adult-specific stem cells that we utilize and we isolate. So one among the such kind of studies, what we have identified is that CD11B negative, TER119 negative means the cells from the bone marrow. When we harvested the cells, we identified them with the type of cells which are negative or null for expression of CD11B or TER119, which is one of the monocytic marker or the ectocytic markers, which is null, but they are positive for CD11. So what we do is that we isolate out. In my next subsequent slide, I will show you a very method how we do this magnetic activated cell sorting, where we incubate the cell, isolated bone marrow stem cells with a particular antibody marker, which the antibody is associated with coated with a magnetic base. So when we pass it through the magnetic columns, we actually isolate out the positive fraction which remains attached to the column and the negative fraction which gets flows out. So similarly, the double negative, what we have identified, the CD11B negative, if I can if you guys are able to see the cursor here, the CD11B negative TER119 negative cells were characterized, and whereas we have taken the CD11B positive and we have selected another populations from those positive populations as CD133 positive. That means the double positives, which are CD11B positive and CD133 positives, and the CD11B negatives are TER119 negatives. We hypothesize that the double negatives are the populations which are differentiating, which can be differentiated into mesenchymal lineages, whereas the double positives are the populations which can be differentiated into the blood cell lineages. And here in this particular figure, what we can see that the double negatives, we are able to be differentiated into osteoblast, adipocytes, and chondrocytes, which are the three typical tri-lineage capacities thumb rule for testing out any mesenchymal stem cells. And of course, the double positives could actually be differentiated into various different types of blood cells, like the CFEP or the granulocyte marker, granulocytes, or the various different types of cells. And not only that, we did also characterize them by expression of the various different mesenchymal markers or the hematopoietic markers in these two different types of cells using at the RNA level or at the protein levels. I'm not showing up all these data out here, but because I want to focus on to the actual part of my today's talk a little later. But to give us basically the students an understanding about how the studies are being done within the stem cell biology field. That's the reason I'm sharing you with it. So these cells were actually when we subjected these cells to different populations onto cell in vitro systems, we could see that there was a difference in the expansion capacity. There was a lower proliferative potential of the hematopoietic stem cells, or we call it as the hematopoietic stem progenitor cells. They were having lower proliferative potentials than the mesenchymal stem progenitor stem cells, or we call it as the bone marrow derived mesenchymal stem cells. So to have this, to work out on this rationale, what we did, my student Neha, my first PhD student Neha, who has worked out on these, we isolated this in this flowchart diagram. I could show you that we isolated these cells based using the max sorting system, and we observed that these double negatives were lacking CD31, what they're expressing of the nestin podexel CD90.2, and they were differentiating to BMS lineages, whereas the double positives were expressing CD31, expressing CD117, SCA1, PROCR1, and differentiating into HSPC lineages. And upon working out more into the molecular mechanism of why the difference in the expansion capacity of these cells, we understood that there were actually different types of histone deacetylases. These are the epigenetic regulators that are being expressed in a differential manner in these two different types of cells, where we observed HDAC2 was expressing much higher expression levels in the BMSEs, whereas the HDAC3 was expressing higher in the HS species, and we could work it out to identify that these HDACs were actually regulating through cyclins by regulating its downstream regulators on the cyclins and the P287s, and these cells actually subject to the level of different to the level of differential cell doubling time in these cells. This work was published a couple of years back in the Stem Cell Research 2016. But as I said, these different tissues of our body has different types of stem cells. So the other tissues like liver tissues, we identified the different specific markers using different specific markers using this technique, as I was discussing, telling you that this is the magnetic cell sorting system, where we have a very simple system to the magnetic columns. We pass out the cells like in the liver homogenates, which we identified sorted out between parenchymal and the non-parenchymal cells, and from the non-parenchymal population we could sort out CD90 positive and CD133 positive. These are the ones which expresses out into the these are the ones which could differentiate into hepatocyte-like cells, and using a particular study where I will be explaining this study more in details in the subsequent one, where we identified that during any kind of injury, specifically to the liver or any skin injury, in injury system there is inflammation that occurs a lot, and then we designed a model, drug-induced toxicity site using chronic toxicity using hepatotoxic and CCl4, and we observed that at a particular initial phases of the 2 weeks of treatment or the 1 week of treatment with a drug, to these animals, when we harvested the cells from the liver, we could see that there is a more of a initially we could identify the CD90.2 positive, CD133 positive hepatic progenitor cells into these tissues. So this work is actually ongoing, where we are identifying what are these locations, or why not these cells are being utilized for harvesting and repopulating the cells in the liver, in damaged liver, which could be. So that research is actually going on, which which I'm not able to share the data with you, because this is the work which we are presently doing. But like every studies, there are challenges in the system. So the basic challenges of the stem cell transplantation therapies are that either you use the endogenous populations of the stem cells, like as I showed you that these endogenous positive cells during the tissue injury, they get repopulated into the tissues, like CD90 positive, CD133 positive hepatic progenitor cells in the liver case of injury. But these recruitments of this endogenous stem cells are very limited to it. So when the injury persists for long, for a chronic kind of injury, these endogenous stem cells are unable to regenerate the damaged tissue. So what the alternative approach to it is transplantation of an exogenous stem cells. Now, how do you identify these exogenous stem cells? As I said, there are specific markers of this. One has to identify and specify, purify, isolate these ones, purify the stem cells based on the specific markers, and then deliver it at the injury site. Now, the question is, the biggest challenge I did, how do we deliver the stem cells? Because when you deliver the stem cells in intravenous route, it goes to the multiple organs of the body, and then it actually very little fractions of it goes to the site of injury. So if the injury is superficial or on the skin of your body, one can utilize it using a delivery vehicle. Now, these delivery vehicles can be any kind of biopolymers or any kind of other cell materials. So one such kind of delivery vehicle is that. One of the biggest challenge that we have to identify an actual delivery vehicle. So these delivery vehicles that can take your cells and get it delivered at the site of your injury, that is increase the engraftment of these delivered stem cells. Or if you are able to deliver the stem cells at the site of injury, the next challenge comes is that because your injury site is mostly harvested with a lot of harsh tissue microenvironment. The injury microenvironment has got a reactive oxygen species, and a lot of other reactive nitrogen species comes to the sites, and then there are a lot of immune activators are there that actually does not help the cells to survive there. So even if you transplant the cells at the site of injury, so next question comes is that whether your transplanted cells are able to survive there, or as well as they are able to proliferate at the site of injury. So this is the next challenge. The next challenge to it is that even if they are proliferating at the site of your injury, it is of no good until and unless they differentiate, or the transplanted cells are differentiating into tissue-specific lineages. So differentiation is the next challenge. So this particular these are the three different challenges. Apart from it, there is a big, as I mentioned, the first different challenge is that very insignificant number of transplanted cells, stem cells, actually migrate or engraft the tissue. Say, for example, you are delivering it into in a therapy mode for a soft tissues which is there inside. You cannot deliver it from the outside of the body, so you have to deliver it through the intravenous route. So the intravenous route, when we deliver it, we have seen that the most part of your cells actually goes and resides in the lungs of the body. Very little of it actually goes to the site of the injury. So how to improve, so my lab actually focuses to answer each and every aspects of these challenges and to deliver a more translational therapy, or more translational therapy that can help to see the light of the day when practiced with the patients. So I'll briefly describe about some of these these words, what we have done, like when we address this challenge of how to differentiate, identify a delivery vehicle for the transplantation in the injury site. We all know that the stem cell world market is improving enormously right now. It's actually growing very fast, and then there are very little of biological scaffolds that are available. The majority of the scaffolds are biological scaffolds. Say, for example, a dead tissue from the cadaver one takes it. So there are ethical issues also attached to it. But there are very little information known about the custom designed stem cell delivery vehicle. Almost they are not yet been practiced in the clinical. Very little of it there in the clinical. I'll share with you some of the information that we are actually being working on. So what we developed is that in collaboration with one of my collaborators here, Dr. Prithvi Vashak, he's a material chemist, and we developed a biopolymer, and that polymer using a specific type of, this is a biocompatible, biodegradable polymer. So these polymer, when we utilized it for transplanting the cells, what we did, we first of all identified this when our bone marrow derived stem cells were cultured along with these porous polymers. This polymer, they actually not only remain attached to the polymer, but they are cultured. They are being observed to be proliferating in the presence of the polymer. That is, the polymer is biocompatible, as well as these biopolymer. We could actually see there were when we sectioned out the biopolymer, after that our cells could actually penetrate inside the biopolymer. So the question was how these penetrations were happening. We identified that there is a mechanism that is actually happening through the activation of ERK-AKT activation, and through MMP13 and MMP2 mediated. They are able to secrete these collagenases, and these collagenases actually what it does, it degrades out the stem's delivery vehicle, as well as they are proliferating within it. When you transplant the cells along with these particular porous polyethylene glycol polyurethane biological scaffolds, they are able to sustain the harsh microenvironment which are present within the site of the injury. And what we did is that we transplanted the cells along with the scaffolds and without the scaffolds, and we observed that the healing time of in a mouse model of wound. This particular, I also like to add that this particular model what we developed is normally in a wound model, in a rodent model, it heals by contraction of the skin. However, what we did is that we have done a splinting model where the splint has been attached surgically put along with this. This is what I'm trying to show it in the cartoon, that this is a splinted model. This is a splinted excisional splint wound healing model where what it does, it replicates the same kind of healing pattern what happens in human body, wherein the facial matter is actually culture. It grows out. The cells come into the play. The inflammatory mediators or the inflammatory cells, as well as that are being, as well as the endogenous stem cells or the exogenous stem cells that we are harvesting it, that remains there, and then they develop layer by layer. The skins are developed. So we could see that in our model, it actually healed the reduced the healing period by 50%. That's a lot of time. That's a lot of good activity that we have observed, and this particular one is not only healed but then also we can see in this particular figure that in the last group, which is the along with the sorry about that, along with the scaffold with PGPU and bone marrow stem cells, which we developed a scarless healing. Now this is happening because it's having a lot of collagen deposition occurring. It increased the collagen deposition. In this particular one, when we cultured the cells along with this scaffold, or when we transplanted the cells, we could see there was a decrease in the pro-inflammatory mediators at the site of injury, increase the anti-inflammatory mediators, and then it led to a wonderful epithelialization happening, as well as endothelial cells formed. The blood vessels, we could observe that could come into the site of injury, and then that healed the skin. And skin regeneration happened without any scars. That is wonderful thing, because we do not want a scar to happen on the wound at the site of your skin, so that can be of more cosmetic and aesthetic values too. So I would like to share with you that this particular our scaffolds has been submitted for patent, and in a very short period of time we could get the US patent granted on this. And because they were we could not find, and neither the patent authorities could find, there were not much of information existing, or there are specifically custom designed stem cell delivery vehicles that are being built. So not only that, we are already into the not only UCB Pharma, which is a Belgium-based company, but also with the other world market leaders of the wound healing companies are already in talking discussions with us for they are showed their interest in our particular patent, and they are utilizing these. They are taking this one forward for clinical trials for this in future. I'd also like to add that particularly this work was highlighted. This was published in Biomaterials by this first author of my work is that Satya, who's my second PhD student, who has worked on this. We published in Biomaterials quite some time back. And this work was actually highlighted by Swissnex, which is the Council General of Switzerland, as one of the top 10 skin research outputs from India. So coming to the next particular challenge, as we discussed, if the cells that we are transplanting are able to comply by the harsh injury microenvironment. So what we question ourselves that as we are using the bone marrow derived mesenchymal stem cells or the hematopoietic stem and progenitor cells, we know that in the bone marrow, every tissue has got a specific site where they are holding these cells or tissue specific stem cells. So that is called the niche of the tissue. So bone marrow stem cell niche has got where these bone marrow derived mesenchymal stem cells are harboring, or the hematopoietic stem cells, or endothelial progenitor cells, or skeletal muscle progenitor cells. There are several different types of tissue specific stem cells which are present in the bone marrow. And at the bone marrow niche, we know that because of hypoxia, there is a ROS. But this ROS is of mild quantity, mild level. However, in a tissue environment, say for example in a skin injury environment, there also contains a lot of ROS, and that ROS is a very bolus ROS. That is a very high amount of ROS. So what we did is that we simulated this in an in vitro conditions first, and we utilized a low concentration. We exposed these cells at a low levels of ROS. As well as high levels of ROS. That is using a hydrogen peroxide, which is a very common reagent that is being used in a cell biological. So this reagent generates a lot of reactive oxygen species. So what we did, we treated the cells with different levels of the hydrogen peroxides, and we observed that we worked out the whole signaling mechanism, what is happening within these cells, why certain of these cells are able to retain and proliferate in a low levels of ROS, whereas it does not. So we identified that when you expose the cells in presence of low levels of ROS, it actually activates JNK pathway, and these JNK gets phosphorylated, which in turn activates FOXO3A. FOXO3A is a transcription factor, which what it happens is there, it translocates within the nucleus and it acts as a transcription factor. And we have observed that in our cells, it is actually increasing the transcription of a particular antioxidant gene called catalase. Now, what does catalase do? Catalase actually reduces it breaks down the hydrogen peroxide into H2O and the other oxygen species. Now, not only that, but we observed that these cells were also getting activated AKT pathway, and these AKTs, which we have observed that downstream, it actually activates cyclins, and these cyclins are helping the cell proliferation. So what we question is that now, if we utilize these particular stem cells, which are preconditioned with a low level of ROS, where we observe that at low level of ROS, these cells are able to proliferate. So at low level of ROS, if we are exposing, preconditioning these cells and transplanting it in our excisional splinting wound healing model, what happens to that? So what we did is that we took this BMSCs, which is a bone marrow derived mesenchymal stem cells, and the hematopoietic stem progenitor cells, and we exposed them to low levels of ROS, and also we did separately as a high level of ROS, but those cells did not survive. But the low level of ROS, when we transplanted preconditioned cells to the site of injury, we could see that again there is an increased proliferation by Ki67 proliferation. We could see that the transplanted cells, what we did is we actually leveled the cells with GFP leveling by expressing GFP onto the cells, so that we can actually distinguish between the endogenous stem cells versus the exogenous transplanted stem cells. And we observed that the exogenous transplanted stem cells were actually proliferating. They were not only that, there were increased reepithelializations in the MSC bone marrow derived MSC transplantation group, whereas the hematopoietic stem cell transplantation group, we could see more of angiogenesis happening. And again, it healed the cells, it healed the transplanted wounds by almost 50% faster. So this particular work was done by Neha, and we published it in ARS as a cover page article. It was a wonderful one. So this study offers a therapeutic approach for improved wound healing by preconditioning the stem cells, which equips them with a better antioxidant defense system. And actually, I would like to share that this is particularly one of the strategies which like not only our group in India, but there are other groups too, are considering preconditioning of a stem cell with particular different types of drugs or systems to equip the cells more robust while we transplant it. So this is particularly coming up, and we are being cited very well for these particular work into the literature. So answering out of this, as I said, there are these cells when we transplant them, if they remain as a stem cell, is of no good. So what we are trying to do identify is that the transplanted stem cells has to differentiate into, that is the third challenge that often remains there in the stem cell biology, is if your differentiation of the transplanted cells into tissue specific lineages. So what Satya did is that we utilize that particular liver injury model, and in the liver injury model, what we observed that in the liver there is an inflammation happening, and this inflammation leads to COX-2 generation, which in turn activates IL-17A, and which also activates the F480 macrophages. In turn, we could see there are a lot of interleukins and inflammatory mediators that are secreted there, and that leads to the apoptosis of the transplanted stem cells, bone marrow derived stem cells. So what we did is that we utilize a celecoxib. Celecoxib is a banned drug, we know that. But what we wanted to identify is that if celecoxib, which is the inflammatory blocker, could actually reduce the inflammation and help these cells to transplant, heal the wound tissues. Interestingly, what we found that we could see when we utilized celecoxib, there was a decrease in various different interleukin 17 level, interleukin 17 level, there was decreased macrophages at the site of injury, and there was an increased survival of these bone marrow derived stem cells. And because of this phenomena, what we have also observed into that in presence of an anti-inflammatory drug, our bone marrow stem cells could show us in vitro level as well as in vivo level too, that they were differentiating more faster into keratinocytes. And that led to increased engraftment of the engraftment at the wound site and reepithelializations mediated wound tissue repair. So this work we published a couple of years back in Cytotherapy, and to share with you that we published in Elsevier publishing house journal, Cytotherapy. The journal themselves highlighted our work in Science Daily on the July 20th issue of 2017, as well as a week later, they have published it in Asian Scientist journal about this particular, because in another word, we wanted to show that this can be utilized as a drug repurposing, where we are not actually don't want to utilize the anti-inflammatory drug to the animals, but we are preconditioning the cells also with the anti-inflammatory drug and then transplanting it to the cells. That is inducing more of differentiation of the tissue-specific differentiation of the stem cells and then healing. So we questioned ourselves: a wound is not normally healed, or skin regeneration can happen not only because of keratinization or epithelial cell reepithelialization, but it also needs a blood vessel formation. So how does this happen? So is there any role for anti-inflammatories to induce this endothelial cell formation. So what we did, my next work, what my fifth student Komal, what she did, that we checked our hypothesis in using a endothelial induction into the endothelial cell formation. So what we did is that we utilized human endothelial cells, which are the Waters jelly-derived mesenchymal stem cells, and we transdifferentiated it in towards endothelial. Because I would like to also mention that the MSCs or mesenchymal stem cells are the unique stem cell population, adult stem cells, which has got transdifferentiation capacity. Means differentiation is normal lineage-specific differentiation, which for MSC is a trilineage differentiation, which is a chondrocyte, as I showed you, chondrocyte, adipocytes, and osteoblasts. But this mesenchymal stem cells poses a plasticity phenomena wherein it can be made into hepatocytes like hepatocyte cells, or it can be differentiated into endothelial cells, as we have done it, as well as others have shown it to be helping in generation of a cardiac cells, but cardiac muscle cells too. So what we tried to show in this particular piece of work, where VEGF receptors are expressed in these induced endothelial cells, and when in these cells actually activate downstream of VEGF receptors, there are different pathways like ERK pathway or STAT3 pathway or SRC, which helps in the various physiological phenomena like proliferation, neovasculinogenesis, and migration. So what we did is that we induced these cells, transdifferentiation of the bone Waters jelly-derived mesenchymal stem cells, human mesenchymal stem cells, into induced endothelial cell populations. We will be calling as the induced endothelial cells, and in these cases, they follow up when we transdifferentiated, they followed into different lineages over the period of time. Early endothelial cells versus late endothelial cells. So when we checked our literature, we correlated our work with the literature, we could find that our cells, that the EC day 14, or we called as induced endothelial cells till at day 14, they very well has a similarity index with the late endothelial cell populations, where these endothelial cells not only differentiate into mature endothelial cells, but they also secrete certain factors, as well as it helps in the endothelial blood vessel formation. Whereas as compared to the early endothelial cells, which we could see in our transdifferentiation phenomena at a very early stage of 7 days transdifferentiation, we could observe that they only has a secretory phenomena, which is called paracrine secretions. It does a lot, but does not differentiate into late stages endothelial cells. So what we did is that we utilized these late endothelial cells, or the ECK14, in our own model, and we transplanted these endothelial cells, induced endothelial cells, and we could see, as compared to the MSC transplanted groups, our endothelial cells transplanted, induced endothelial cell transplanted group could show increased vascularity formation. Now, what we did, we questioned ourselves if we can use our preconditioning model that we have shown earlier of celecoxib, which is a COX-2 inhibitor. What we did in presence of, while inducing the cells into induced endothelial cells, we blocked the endothelial cells. When we blocked the endothelial cells, we could see that it activated particularly certain groups, that is a phospho-STAT3 group, or ERK, as well as SRC. It activated it. So downstream, it activated this particular phenomena, and that transplantation of this preconditioned celecoxib preconditioned endothelial cells potentiated wound healing by increasing the vascularity. So this particular work Komal published it in Cytotherapy in last year, and we have shown that these transplantations of the induced preconditioned cells, they avoid one of this phenomena, the non-targeted effect of detrimental effect of the celecoxib. As I said, that celecoxib is a banned drug because it causes a lot of cardiotoxicity as well as hepatotoxicity, so it is not being used clinically. But celecoxib, what we did, we exposed, preconditioned the cells. We did not inject the cell drug into the animals, but what we did, we preconditioned our cells in presence of celecoxib and transplanted the cells, preconditioned cells, and we could increase the endothelialization or the neovasculogenesis at the injury site, and that helped in the mediated skin tissue repair. So when we answered these questions, that the challenges of how to deliver stem cells for skin tissue regeneration, how to increase the proliferative potentials, and how to deliver this, increase the differentiation potential too. Then the next question comes, how do we increase the engraftment capacity utilizing a therapeutic mode? That is, when we transplant the cells through intravenous route. So what we did, we questioned ourselves, how can we improve this the engraftment of a transplanted cells? We know that there are several different types of chemokines that are expressed at the site of injury. What we did next, we evaluated various different types of chemokines and their cognate receptors. What we did, we performed an array of microarray kind of analysis at the wound site and the wound region temporarily from day 0 to day 10, and we observed that there are among the various different types of chemokines, CXCL16 showed a persistent increase right from the day 0 to the day 10 level, and the wound in the excisional splinting wound of a mouse model, it showed an increased level of CXCL16, which is a chemokine. So we questioned that. These chemokines we know has a cognate receptor, and these cognate receptors, we question whether or not these cognate receptors are expressed in any of our stem cells, adult stem cells, say for example, mesenchymal stromal cells. So what we did, that we utilized this, compared it with the bone marrow derived stem cells versus mesenchymal stem cells, and we observed that various different CCR3, these are the receptors for CCR3 or CCR7 there, as well as CXCR2, CXCR6, CXCR7. What helped us to show that, understand that CXCL16 is a chemokine ligand that has been expressed in a large amount at the site of the injury. Its cognate receptor is CXCR6, and we observe that our MSCs do express a lot of higher concentrations or higher levels of CXCR6. So we understood that there might be a certain kind of phenomena if we can transplant these cells, and because of the wound site having its cognate ligand, these cells expressing the receptors can definitely migrate to the site of the injury and can help. So what we first did is that we wanted to study at the in vitro level what happens in presence of these particular ligands. So we evaluated the MSC physiology, like the proliferation or the migrations. We observed that using various different types of readout techniques, like the proliferation BRD incorporation assays or the coliforming unit assays, we could not see any effect of CXCL16 onto the MSC's proliferation. So the question next we did is that we wanted to study the migration. And to our surprise, we could see that at a significant level, at a 10 nanogram per ml level in an in vitro, which is a Boydin chamber assay, what we have done, chemotaxis level, we could see that at a very low level, at 10 nanogram per ml, our cells could migrate to the lower chamber. So what we did, we wanted to see whether this is a very genuine effect that's happening. We utilized a CXCL16 neutralizing ligand. In presence of the CXCL16 neutralizing ligand, we could see that there was a dose-dependent increasing dose-dependent of CXCL16 neutralizing ligand. We could see a decrease in the migration level of the cells. So that proves that yes, it is happening through the CXCL16 CXCR6 axis. That is, the migration of these MSCs are happening through this CXCL16 CXCR6 axis. So next, we wanted to see in an in vitro level what are the molecular changes that is happening. That is a signaling mechanism. We evaluated directly the migration pathways, the markers of the modulators that are the effectors that get modulated in presence of during the migration, like phosphophate. Yeah, we could see that in presence of the CXCL16 ligand, we could see there was an increased phosphorylation activation of fat, SRC, as well as phospho-ERK. So we hypothesized that in presence of the CXCL16 ligand, which binds to CXCR6 onto this receptor on the MSCs, that in turn activates fat, which in turn activates SRC, which in turn activates ERK. And we know that ERK, when it gets activated, it translocates into the nucleus, and in the nucleus, it acts as a co-activator along with the other transcription factors who activates its downstream target genes. So here we hypothesize if the cells are migrating. So let's hypothesize that if MMP within the MSCs, MMPs are getting activated, and these MMPs are actually helping the cell MSCs to migrate. So to prove our hypothesis, we performed a battery of experiments where we first of all evaluated the expression levels of the MMPs, like MMP2, MMP9, MMP13, which are the collagenases, the gelatinases, where we observed that among all these, MMP2 was significantly increased not only at the RNA level, but also at the zymography, where we know that this is the proteins which are expressed in the PPO levels. So they are one in the E figure, where we've shown that very systematically that whenever we are utilizing these blockers, till before the blockers, like in presence of the FAC14, we could not see activation of FAC, but in presence of SRC blocker, we could see activation of FAC, but not the SRC blockers. And if you can go through this figure very carefully, you can appreciate that individually each and every blocker could activate these particular blockers, could activate all these particular transcription, all these particular immune FAC, SRC, and ERK. However, when we inhibited with the MMP blockers, we could see activation of all these particular markers, but not the MMPs. So what we did is that what happens when you overexpress CXCR6? Is the effect much increased, much higher? And to our surprise, we did say that, but we used a plasmid CMV CXCR6. We transfected the cells with these particular plasmid CXCR6, and we could see that there was a marked increase in activation of these particular signaling pathways, and also there were reduction in presence of the blockers, but to a limited level, because we are overexpressing CXCR6. But MSC migration. Now, when we, as I mentioned, that ERK is one where it is getting translocated into the nucleus. So we wanted to prove that whether or not it is happening in our system. Yes, we did see that in presence of CXC60, what we did when we utilized a phospho-ERK using immunocytochemistry, where in the cells, we could see that all of the ERKs are located into the nucleus, which is being co-stained using the DAPI, which is a nuclear stain model, nuclear stains, and we could see there was an increase of CXC16 induced expression, in presence of CXC16, phospho-ERK is translocated into the nucleus. And to our surprise, when we overexpress CXCR6, this nuclear translocation was much farther, much more markedly increased. And this blocker, using these various different blockers, we could see that all these cells, the ERK translocation has happened, except GM6001, which is a MMP blocker, pan-MMP blocker, where in presence of pan-MMP blocker, we could see ERK getting translocated into the nucleus, but migration did not happen, because MMP is downstream to ERK, where it gets translocated into the nucleus. So we question ourselves, when ERK is getting translocated into the nucleus and can activate as a co-activator, co-transcriptional activator, along with the other transcription sites, what are those transcription sites that are present onto the promoter region of these MMP2 that can be maybe getting activated? And we could see that SP1 binding sites were observed, 3 different SP1 binding sites were observed onto the MMP2 promoter, as is shown by the schematics. So we identified into the literature if there was any role to play with phospho-ERK getting activated and binding to the SP1. And we did observe that, yes, there are reports about phospho-ERK when they get it has got a binding capacity with the activated SP1, and that in turn activates various different transcription of different target genes, but it was not known about the MMP2. So what we did, we generated MMP2 promoter constructs, deletion mutant constructs. One is a wild type construct which had all the 3 different sites, and then there were constructs which were being deleted out individually, and we observed that in presence of wild type MMP2, in presence of CXCL16 or CXCR6 overexpression, there was a significant increase in the promoter activity of wild type MMP2 promoter, which was decreased in presence when we deleted out the BS3 site, and furthermore deleted when we decreased both BS3 and BS2 site. But when we deleted this BS3, the BS1 site, but had this BS3 and BS2 site, we could see that the promoter activity was negative, increased high, but not as significant as a wild type, thus suggesting that among all the 3 BS2 and BS1, BS3 and BS2 sites are the important sites for binding of activated ERK along with the SP1, co-activating through SP1, but nevertheless all the activated sites were important. So what we did in presence of the ERK blockers, we wanted to show that yes, in presence of ERK blocker, the luciferase activity went down, and to furthermore make it understand whether or not it is happening through ERK, through CXCR6, what we did is that we overexpressed CXCR6 as well as we silenced our CXCR6. And in presence of silencing of CXCR6, we could see there was a decrease in the promoter activity. And furthermore, what we did, we utilized a chip analysis, chromatin immunoprecipitation assay, where we utilized that in presence of CXCR6 overexpression, we could see that there was an increased binding at the BS2 and BS site, which correlated very well with the promoter activity assay, thus telling us that CXCR6 and signaling activation helps for the nuclear dislocation of phosphoreactive, and that in turn activates, regulates MMP2 transcriptionally, both at a promoter level as well as at a mRNA level. So next question comes: what, after having understanding the whole of the in vitro signaling mechanism, can we not develop our models? So what we did, we utilized a stem cell gene therapy approach, as my title suggests that. So what we did, we on the left side of this particular very busy slide, where we have shown that we subcloned from the plasmid construct, the CXCR6 gene, we subcloned it into a lentiviral construct. Why lentiviral? Because lentivirals are the kind of viruses which can transduce not only the dividing cells but also the non-dividing cells, and thus you get more and more expression of your transgene. So what we did, we utilized these overexpression systems, and using a standard molecular biology tools, what we have subcloned them, and we did a sequence analysis, and we observed that the sequence correlated well aligned with the CXCR6, reported CXCR6. So what we did, we developed the lentiviral particles using a HEK293 T cells. And these lentiviral particles, as it shows, that again I would like to address here is that our construct also had the GFP. Okay, so GFP along with the CXCR6 that helps us to locate the cells when we utilize it not only in the in vitro systems under the microscope but also in the in vivo systems at the site of the injury or at the site of its application. So what we have done, we observed this when we transduce these cells, our MSCs along with the lentiviral constructs, lentiviral particles overexpressing CXCR6, we could see there were a lot of these cells, all the MSCs, and that what we utilized using our prior standardized model in our lab is the excisional splint wound healing model, where we transplanted the cells not at the site of the injury through intradermal route, whereas we did it through the intravenous route, through the tail vein. When we injected the cells through the tail vein and we could see there were a location of these cells going to the site of the injury. And to our surprise, what we wanted to have another group where we actually increased the level of CXL16 at the site of the injury by giving a very localized injections of CXL16 at the site on the wound site, and we could see that there were much getting into the site of the injury. So this study was done in a very elaborated way in 3 different types of animal models, where we not only used the excisional splint wound healing model, normal model, but also we questioned then that why not we utilize it in a diabetic model, where we know that in diabetic patients they develop foot ulcers where the injuries or the wounds does not heal because of the high levels of glucose into their body. And so we developed 2 different types of diabetic models in our lab. One is the type 1 diabetes using a streptozotocin, which I will be explaining you in our subsequent slides how we do it. And then another one, we use the transgenic models, which is the DBDB mice, where we utilize we take the transgenic mice and then we inject it through the tail vein, our cells. So in our model, we could see that in the streptozotocin-induced type 1 diabetic model, as well as the type 2 diabetic DBDB mice, in the presence of the CXCR6 overexpressing cells, could show a significant increase in the wound closure. The percent wound closure was much higher as compared to the non-transplanted, as compared to the empty vector transplanted cells. So we could increase along with the we could decrease the wound healing time and increase the wound closures. And as the right hand side picture shows, in the cytochemistry, where the cells and a higher magnification, you could appreciate that the CXCR6 expression levels were seen along with the GFP. As I mentioned, if you remember, our cells had a construct along with the GFP, our construct had a GFP along with the CXCR6. So GFP expressing cells, we could see there were, to distinguish between CXCR6 expressing the endogenous cells, we could distinguish them from the transplanted group. So these transplanted cells are actually getting engrafted at the site of injury and helping into the wound healing procedure. And this work was done by Neha, and we published this year, a couple of months back, in Molecular Therapy. It's a very high regarded journal in our field, where we showed that the therapeutic effects of our CXCL16, CXCR6 signaling axis, which can enhance the CXCR6 mediated enhanced homing of our transplanted cells at the site of the injury. So, utilizing this particular technique of stem cell gene therapy approach, we wanted to question that when we know that, as I was mentioning a couple of slides back, that neovascularization is an important phenomenon. we could see there were vascular cells also growing into the site of the injury in our this particular model too, where we have transplanted MSC, CXR6 MSC gene therapy. But we wanted to question if we can increase more of vascularity, if the healing pattern helps much more faster in the diabetic mice. So what we wanted to see is that what is the information or exists about that. As I mentioned about that, neovascularization is a crucial step for wound healing purpose, and endothelial progenitor cells, which I would like to introduce under the term EPCs, as I was telling in the earlier slides, EPCs are the one which gives rise to the endothelial cells upon maturation and differentiation, and these cells play a very vital role in healing of a wound if endogenous progenitor cells are effective enough in their presence. However, we know that in a diabetic patients, these EPCs are compromised and they do not help in the healing of the wound. So they develop diabetic foot ulcers. Now, apart from that, recently we reviewed this and we identified that in a clinical stages, apart from debridement of the wound and application of the antibacterial or anti-inflammatory agents, the delivery of pro-angiogenic factors has been practiced. It's an FDA approved process where among the different types of growth factors like VEGF, PDGF, or TGF, or FGF, this Regnerenix, which is a becaplermin, it's a hydrogel-based polymer, which actually has a PDGF growth factor that limits the growth factor utilization into the patients. So what we did is that we wanted to question ourselves, what are the different methods, as we know, which are existing in the development of endothelial progenitor cells. Now, if we see in this cartoon below, the ECs are actually endothelial progenitor cells get matured from the endothelial cells get matured from the endothelial progenitor cells, or from the embryonic stem cells, or from the very small endothelial-like cells, or the amniotic fluid cells, or various different other types of cells. Among the several different strategies that were explored to induce this endothelial proliferation or endothelial cell generation, one of the type of cell is the mesenchymal stem cells. So we have been utilizing this model. Now, not only that, our lab loves to work on MSCs, but MSCs are abundantly available in the bone marrow. And I explained to you that MSCs has a unique property of plasticity. That means they can be transdifferentiated into endothelial cells or hepatocyte cells or cardiac muscle cells or any different types of other cells. And we have already earlier in my presentation, I've showed you our lab has developed the data how we have induced endothelial cell generation in using a MSCs. So what we try to identify these particular cells and utilize a different model of understanding how neovasculogenesis mediated tissue rejuvenation can be developed. What we did is that we questioned that in literature what exists. We performed an in silico analysis using the existing microarray data sets that are available in the literature. We harvested these particular data. We processed them, normalized them from 3 different population cells. One population of cells is the human mesenchymal stem cells. Another population is the late endothelial progenitor cells, as well as another is the HUVEC, which is a mature and human umbilical vein endothelial cells. We utilized these 3 microarray data sets from the literature, and as is shown, and we cured this particular curing, and we identified from these particular different sets using in silico analysis techniques, which I'm not going in detail about it. We identified several different genes which are being induced, upregulated, or downregulated in these cells during these cells. Now, what we identified the similar, which are those common genes which are actually getting upregulated or downregulated. Say, for example, a gene which was observed to be upregulated, increased expression was higher in MSCs, but in EPCs it was less, and finally in mature endothelial cells it was further less. Or in MSCs which was less, in EPCs it was more, and then further in mature endothelial cells we could see much more. So we identified those bunch of cells and we cured the cells as shown in the PCA plot analysis. The populations of those particular microarray data sets what we have utilized, they were very intact, that means showing the less variations, but they were very far apart from individual different types of, like the MSCs were far apart from the late endothelial progenitor cells were far apart from the mature endothelial cells. That shows that the curing of the data sets of the in silico data sets were very very successful and were done very relevant, very high. And now what we identified, there were a bunch of different types of genes, target genes we could identify from these microarray datasets, meta-analysis of these microarray datasets. Now, what we did the heat map, and we identified which are the genes which are overexpressed, and then we did a complete array of this, of all the genes using a quantitative real-time PCR analysis, and we observed that from the human MSCs to the induced endothelial cells at the early time point, like early endothelial cells versus late time point or the late endothelial progenitor cells, there were differential expression of these particular genes. And we could see that in the differential, there are lots of these angiogenesis related genes. I must add to it, we focused our array datasets, compiled the datasets from there, and identified those genes which are related to the angiogenesis pathway. So we identified those angiogenesis genes, and we could see that there were a lot of genes which are getting upregulated, and a lot of genes they were also getting downregulated. So we confirmed our microarray datasets or microarray datasets using our real-time PCR analysis. Not only that, for these particular microarray datasets, we identified in silico validated data, and we particularly put these particular target genes, made hypothesis, and put these target genes in a string software. In string software, we get to know from the literature how many of those genes are relatively known or cured, or they have been experimentally worked out among these. So what we identified, we took BMP2, SMAD1, KDR, SFRP4, ID1, twist1. Why? You will be coming to know in my next model why we tried. And among this, we could see there were a lot of information existing in the literature from BMP2 through that acting upon through SMAD to ID1. And very little was known about through KDR or twist. But there were no correlation between twist to KDR or twist to SFRP4. Twist1 is a transcription factor. Now, what we wanted to know, and so is the ID1. So what we tried to hypothesize here in the model, BMP2 is a growth factor, and this particularly binds to its receptor called BMP receptor 1 or receptor 2 onto the surface of the cell. And in MSCs, what we have studied in our model, MSCs, it is expressing in MSCs. So it binds to its receptors, which in turn activates SMAD1 by activating with. Now SMAD1 is a transcription factor. What it does, it actually so this trimeric, they get inhibited in presence of DMH1, which is a receptor blocker of BMP receptor 1. And with this trimeric complex, SMAD1, SMAD5, and SMAD8, in turn recruits its SMAD4, which helps in translocating this trimeric complex, that's 4 transcription factors together into the nucleus, and where SMAD4 recognizes the binding site onto its transcription, onto its target genes. Here in our case, we say that it is an ID1. ID1 stands for inhibitor of differentiation. Now, in MSCs, it activates and it expresses out ID1 gene. All right. Now, what is happening? What we hypothesize? What is happening during our transdifferentiation? During the transdifferentiation, there is another 2 types of SMADs, which is called SMAD6 and SMAD7, which are inhibitory in nature, and in literature it is known. Now, we are proposing that SMAD6 and SMAD7 is getting activated. Its expression goes higher. That in turn inhibits the recruitment of this SMAD1, SMAD5, SMAD8, SMAD4 complex to the nuclear translocation. When it does, it inhibits the nuclear translocation of this SMAD complex. It inhibits the expression of ID1. Now, when it inhibits the expression of ID1 is an atypical transcription factor. What do we mean by atypical transcription factor? Atypical transcription factors are the ones which do not have a DNA binding site. But then how does it work? It works through binding to other core transcription factors, that in turn helps in downstream regulation of its target gene. So in our case, we hypothesize that ID1 is a basic helix transcription factor. ID1 is the atypical transcription factor, which has a binding tendency for binding to the basic helix class of transcription factors. Among the basic helix class of helix transcription factors, we could identify there are A1, there is a known to shown in the literature to be one. But we hypothesize that ID1 here in our case must be binding to twist. Why does it so? Because twist 1, we could do in silico analysis, and we could observe that during transdifferentiation, when ID1 is expression goes low, it do not bind to twist 1, and therefore twist 1 is available freely, which in turn binds to its target gene called KDR. Again, I would like to add here is that twist1 is a kind of transcription factor which has shown to actually activate its downstream target as well as inhibit its expression of its downstream target. So we hypothesize that there are 2 genes which we identified to be essentially required for endothelialization. That is called as, we call it as KDR, which is a VEGF receptor 2, and SFRP4. Now, what we hypothesize that twist1, which is during transdifferentiation, is freely available, not bound with the ID1, helps in binding to the KDR site, but twist1 binds onto the EVOX regions of the on its target genes, and it transcriptionally activates KDR as well as transcriptionally downregulates SFRP4. So, with this particular schematic of a plausible signaling mechanisms during the transdifferentiation of MSCs into EPCs, we particularly generated a model. What we did? We utilized first to study the in vitro model. And in vitro, what we have shown that the human MSCs, what we done when we transdifferentiated, there's a lot of, we utilize a very standard assay called di-acylated LDL uptake. Now, di-acylated low density lipoprotein is a very it is a characteristic uptake which is done by endothelial cells. So we utilize this particular model to show that during transdifferentiation of our MSCs into induced endothelial cells, we could see a lot of di-acylated LDL uptake happening, which was increased further in presence of BMP2, which is an activator. If you remember the cartoon what I made and I showed, and which is inhibited with the help of DMH1. If you remember, DMH1 is the inhibitor of the BMP receptors, and we could see that not only it inhibited the uptake of, not only increased the uptake of the di-acylated LDL uptake, but also increased various different expression of the endothelial specific genes to add in the transdifferentiated cells in presence of BMP2, and that was perturbed in presence of its receptor block. Having said that, what we did, as we hypothesized SMADs are involved, what we did an array of all the SMAD genes, expression of the array of all the SMAD genes, and from there I could show you only few over here, as that the SMAD along with the SMAD1 and SMAD2, which are expressed, SMAD1 and SMAD6, along with the other SMAD1, which is a pro-activatory SMAD, the inhibitory SMADs, as we hypothesized, SMAD6 and SMAD7 was also increased, expression level was high. So this gives us the idea that BMP2 is potentiating these human MSCs during transdifferentiation into endothelial cells via the differential expression of the SMADs. How does it do it? What we did, in presence of these particular BMP2, we could see that there was an increased expression of ID1 in the MSCs, but not in the induced endothelial cells. So again, I would like to go into that. We could show at a promoter level it is working, as well as at a protein level too, where we are showing it here that in endothelial cells, twist1 expression is much higher at a protein level as compared to the MSCs, whereas the ID1 level is low as compared to the MSCs. And then what we did is that we pooled it using our ID1. That we performed a co-immunoprecipitation assay, and we pulled the ID1 cells with ID1, and we observed that there was an increase when we plotted the samples with the twist1. We could see that there was an increased pull down of twist1 in the MSCs, but not in induced endothelial cells. So that tells us that yes, ID1 is playing a role during expression of ID1. We are able to see that the transdifferentiation of the cells are happening. Now, ID1, as I said, is a nuclear transcription factor. We could also show that in presence of BMP2, ID1 is getting nuclear translocated in the MSCs, but not in the endothelial cells. In endothelial cells, it does not get nuclear translocated. So this further tells us that BMP2 mediated co-regulation is happening of ID1 and twist1 that occurs during the transdifferentiation of our. So partially we are proving our own hypothesis, as I mentioned in our cartoon, at that in turn. But now next we ask the question, as we hypothesized of twist1 having by having binding sites onto the KDR promoter and SFRP promoter, we could show that there are two different binding sites of twist one, that is two different e-boxes onto the KDR promoter, and there are three different binding sites on the SFRP promoter. And among the two different binding sites, through promoter reporter assay, we could show that the binding site one is most essential for binding onto the of KDR twist one onto the site and promoting expression of them. That in turn was inhibited in presence of silencing of twist one. So what we are trying to do, not only overexpress in presence of twist, we did the promoter reporter assay not only in presence of overexpression of twist one, co-transfection of overexpression system, but also we did separately co-transfection of a silencing system to prove that yes, that is what is happening because of the twist one. And so there were also shown that in SFRP4, we could see that there are two different binding sites on the SFRP4 that were very crucial for binding of these particular, for repression of these SFRP4 by twist one. That was perturbed in presence of silencing, but not to the extent. However, we hypothesized that in absence of in presence of silencing of twist one, this could have gone much higher, but it did not. So that tells us that there's this transcriptional regulation by twist one on SFRP4 promoter is happening in part. So we also observed in MSCs, but that level of transcriptional upregulation is not to that extent as compared to the endothelial cells. Next, to prove that yes, this is happening through this binding, we performed the chip assay, and to show that, as we have observed in our promoter reporter assays, we could see that the binding site 2 was, as I mentioned in my previous slide, binding site 2 as is showing much more binding of these particular twist 1 onto the site, and similarly binding site 1 and binding site 2 on SFRP4 has shown to be the prime site for binding of twist 1 and regulating the transcription regulation of these target genes, like KDR, so upregulating KDR and downregulating SFRP4. So what we did, we utilized, so far what we did was a transient transfection using plasmid vectors. Now we question that using our gene therapy approach, human gene, we tried to make a stable cell. How did we do? We particularly overexpressed twist 1 or silenced twist 1 separately in MSCs and then induced themselves towards the transdifferentiation process. Now, when these cells, we observed that there was a shift. These cells were also having, as I mentioned, our expression system, our expression cassette also had a GFP. So the cells which were overexpressing twist 1 were also overexpressing GFP, and cells which were silencing with twist 1 were also having GFP. This is a commercial expression vector that we have purchased and we have utilized. But the lentiviral vector we generated in our lab and we overexpressed. And we generated lentiviral vector system from the plasmid vector system of the plasmids of shRNA also. And we could prove it with immunocytochemical assays as well as the RNA level. There is overexpression of twist 1 as well as silencing. Not only that, we have also shown that in these particularly reprogram cells. Why I say that? So because twist 1 is a transcription factor, we are trying to reprogram mesenchymal cell towards the endothelial lineage. So we introduce the term reprogram cells. So it's reprogrammed endothelial cells. So the reprogrammed endothelial cells would show a significant increase of GFP expressing KDR positive cells, as well as decreasing in low levels of SFRP, low levels of KDR in presence of twist 1 siRNA, and in presence of the reprogrammed twist 1 reprogrammed endothelial cells, there were a significantly higher level percent of cells which were KDR positive. Just the reverse has happened with the SFRP4 along with it. So having these stable cells already existed in our hand, what we did, utilizing our previous developed system, we first of all utilize an in vitro assay where we have shown that it is actually regulating the proliferation, but not only proliferation, but also the migration of it. Migration is increased, and we performed a CAM, choriogenic alveolar membrane assay, which is a very standard angiogenesis assay, where we have, in presence of these particular reprogrammed twist 1 reprogrammed endothelial cells, we could see increased vascularity in the CAM as compared, which was completely negative in the absence of the twist shRNA in our group. And this, we calculated the blood vessels, the length. So this, again, I would like to add is an in vitro system to address the angiogenesis. So the twist 1 overexpression, it's regulating the vasculogenesis related physiological functions, that is increased migrations as well as disqualification. Now we developed our two diabetic model. As I promised you previously, what we did, we have shown that we have utilized a type 1 diabetic model, which is a very standard model using a streptozotocin. Streptozotocin is an antidiabetic drug, but when we been given at a 70 milligram per kg body weight, which is standardized in our lab, we could develop for over a repeated 4-5 doses after in a start animal over a repeated dose of 5 doses in a span of 24 hours. We could develop diabetes for 2 weeks, and that remains persistent for a long period of time, which we have actually confirmed that the blood glucose level is much significantly higher, and it has crossed the blood glucose level of a diabetic limits in a mouse. We developed the wound, so that we considered at day 0, where we generated an excisional splinting wound model into the mouse, and we transplanted the cells intradermally. These cells were transplanted intradermally, and then we, at a regular span of time, day 3, day 7, day 14, we observed the morphological assessment of the wound, as well as finally we harvested the wound and we utilized it for understanding the molecular at a molecular level, the expressions as well as the healing pattern. On the contrary, we also utilized type 2 diabetic model, which is a transgenic DVDV mice. So what we do is that there's a transgenic mice, which is a very commonly used or very known model, and we transplanted the cells by doing the now each model. One group, which was the plain, that is the empty vector control group, empty vector control, which we named as induced endothelial cells, which we are not reprogramming the cells towards endothelial cells. Another group was a twist one reprogrammed endothelial cells. Another group was a control, that is a scrambled shRNA group of induced endothelial cells, and another the twist one shRNA, that is a twist one silencing group, where we are utilizing as a twist one silencing. So there were 4 groups we utilized, and we transplanted these 4 different groups in 2 different types of models, and then we performed the in vivo assays. And what we did was, utilizing a laser Doppler flowmetry, we wanted to identify, we wanted to study the microcirculatory blood flow, and we could see that in our twist one reprogrammed endothelial cell, we could see much marked or significantly higher level of flux intensity, or there is a microcirculatory blood was much more higher as compared to the control, as well as much significantly higher than the silencing group. That is a twist one shRNA silencing. Not only the microcirculatory blood flow, but also the morphometric analysis, as we see, the wound edge, as is shown by here, that showed a significant drop in the wound edge, that is decrease in the wound edge. That means there were much more significant increase in the percent wound closure in the reprogrammed endothelial cell group, the transplanted group, both in the type 1 and type 2 diabetes. Not only that, when we perform the histological assays, not only the HLE but also the collagen deposition we utilized, we could see that there was a decrease in the vertical increase in the vertical epidermal thickness in the reprogrammed endothelial cell transplanted group, and a decrease in the wound gap as compared to the control versus the shRNA or the silence group. Similarly, next, what we wanted to show that in our wound site, whether or not the cells are expressing the transgene, what we have transplanted. If we could see here that our cells were overexpressing twist1 along with the GFP. If you remember, our constructs have the GFP. So what we did, we highlighted, we magnified using in a which were twist1 positive or also GFP positive. That means these are the trans which are getting engrafted at the site of the injury, and which was much more higher as compared to our blood. This was done on multiple different tissue sections and multiple different models, multiple different ends of mice in the. Not only that, it is getting engrafted at the wound site, but then when we question that these are human mesenchymal stem cells that we have reprogrammed them to form human-induced reprogrammed endothelial cells. So what we say that when we are transplanting these cells and if it is getting engrafted at the wound site, are there genes of human-specific genes are expressing out there? If it is so, if the cells are getting engrafted, it is true, then we should be able to get to see the human-specific genes too at the site of injury. So at the site of injury, when we evaluated the human-specific endothelial cell markers, we could see a significant increase in human-specific genes of endothelial cell markers. So that tells us that, yes, our transplanted cells are getting engrafted at the site of injury, and they are getting differentiating their mature endothelial cells and forming the blood vessels. To prove that, we also know that MSCs do not only heal by itself getting transdifferentiated, but there is also a school of thought where they say that MSCs secrete a lot of trophic factors or growth factors, chemokines, that help in these particular factors actually help in the regulating or modifying the microenvironment cells, the disease microenvironment cells or the tissue injury cells back into their phenotype, and then that helps in the cells to grow. That helps the cells to or the injury to heal. Therefore, what question and we wanted to see if at all our transplanted cells are secreting any factor, we performed a mouse-specific gene expression levels at the injury site of these particular wound sites, and we could see that in the reprogrammed endothelial cell group, which are having much higher increase in engrafted cells, could also show that mouse-specific endothelial cell specific genes also. So this tells us that engrafted twist-one reprogrammed endothelial cells are not only healing by itself getting transdifferentiated into endothelial cells, but also helping the wound microenvironment by secreting certain factors and helping the endovascularity at the wound site. Now, our target gene for twist-one was KDR and SFRP4, as I showed it in vitro mechanism. So now we wanted to show in vivo conditions in our wound model, in a preclinical model, these cells were also showing much higher in the reprogrammed endothelial cell transplanted group, much more GFP expressing KDR as compared to the control or the silencing group. On the contrary, SFRP4 was observed to be much higher expressed in the control or the silence group, but not on the overexpression. So this proves that the twist-one reprogrammed endothelial cell transplantation regulates the KDR and SFRP4 expressions even in the in vivo condition at the regenerated wound site. To prove finally that neovascularization is happening, we utilized CD31, which is a very well-known marker, PCAM of vasculogenesis. Now, GFP expressing, we could see that there were a lot of GFP expressing CD31. That means our cells which are transplanting are also expressing CD31. Not only CD31, but we know that our endothelial cells, blood vessels are often forms not only by its own endothelial cells, but also the wrapping around of the smooth vessel cells, which express as alpha SMA. And you can appreciate in our confocal microscopy of a larger magnification, we could see that vascular sections were observed much higher in our twist-one reprogrammed endothelial cell transplanted group as compared to the other group. To conclude from these particular studies, what I have shown so far, that transplantation is neovascularization, microvascular blood flow in a diabetic mode, where the normally healing is very difficult. In an in vivo model, we could see that for months together in a type 1 or type 2 diabetic model, it does not heal with the help of only MSCs transplanted group. But when we are transplanting this, it is healing up by 14 days faster, that is 50% faster. That's a lot of fast in healing. And further in our data, which I have not shared with you here, when we left this animals for a longer period of time, till 1 month, we could see that in a diabetic model too, it heals up completely with our MSC or the twist-one reprogrammed endothelial cell transplanted cell group. Not only that, the transgene, as I said, it not only led to the human-specific but also mouse-specific vascular gene expression, which tells us that the regenerated diabetic wounds, thereby suggesting higher engraftment and paratime regulation occurring at the mediated effect. Now, in vivo, twist-one was also shown to have its target genes like KDR and SFRP4 regulated. But not only that, but CD31 as well as the other vascularization markers are also shown to be expression. But I must also add, like any research studies, this study also has a limitation. What I want to share that we have studied only for a limited period of time, but we have not studied for a longer period of time. Might be for a longer period of time. We have to study to see if there is any case of any malignancies happening or occurring into that. But to share with you, this work was done by Komal, my 5th PhD student, and we published in Diabetes very recently, two months back, and it has got a very wide circulation, and it's a very nice journal in the field that shows. Not only this, my lab also works, which I'm not going to share today because I've already taken a lot of time. Now, we have been working on liver regeneration as well as other skin regenerations. Finally, I have to acknowledge my collaborators. I have not only limited to this, but I've got a lot more collaborators outside India, along with my funding agencies. Most of this work was done by CSR funding, CSR niche creating project, which I was being funded by the third funding Niti Aayog projects which we are funded for. And like to thank, today I've shared with you my work with Neha's work, Neha, Satya, and Komal, but Soumya worked in the liver fibrosis in the past in my lab, and Raju worked on the cancer stem cell. All the 5 alumni of my lab has been working in as a postdoc in various different universities in the US, and presently my lab is with 6 PhD students. Ragini, Digvijay, Surbi, and Trisha are working on the cancer stem cell projects, whereas, as I mentioned, Shubha and Shilpa are working on the regenerative medicine projects. So, as I said, there are postdoc and PhD positions that are available in my lab. If anybody is interested, any students, you can contact me. My email ID is here. I'll be happy to answer any questions. Thank you.
Thank you, Dr. Amitabh Dash, for your nice lecture, nice presentation. We had a very lengthy presentation, and I hope there will be a lot of questions. No, it's okay, and I know that there are a lot of questions, so I would request my colleagues, Sri Montaj Mondal, to kindly take the questions and redirect it to you. Thank you.
Sure. I hope I have been pretty lucid for everybody to understand. Sure, please go ahead.
Thank you, everyone. Due to the time limitation, we are unable to take all the questions from participants. We have sorted out some questions and will convey to our speaker for his valuable feedback. There are so many questions in the chat box of the YouTube live streaming. Sure. We have sorted out some questions and post it in the chat box of the Meet.
Okay.
Kindly read out the questions and give your valuable feedback.
Okay, I could see the immediate question right now by somebody, Shijani Hajra. Stem cell therapy, how much effective in case of blood cancer treatment? I would like to, my lab doesn't work on blood cancer, but then I know there are a lot of work going on. Indeed, there had been some of those work going on in blood cancer, that is the blood bone marrow transplantation occurs in blood cancer. They replenish the blood. Yes, indeed, there is a role to play. And in clinical stages, there are some of the work which is being done to work on this particular field. It is going on. It is actually working out. You know, to give me a very elaborate scientific answer to it, it will take time. So I hope I will be able to answer them all on email. But then other questions, if there are. What is the future of stem cells in our life, like if someone lost their organ? Amrita Kumari. Let me answer you, Amrita. I'll tell you that. Would you like to, Dr. Gautam Ghosh or somebody associated with Dr. Gautam Ghosh sitting over there, would you like to spell out the questions by yourself, or should I read it and answer it?
Yes.
Okay.
Sir, if you can read out question.
Okay. Okay, so quickly I'll answer you this. Yes, definitely there is a lot of future actually happening in presently at a clinical situation. I will tell you that I've met with several surgeons in India also. It is doing what they do is that they, in cases of chronic injuries like a diabetic wounds as well as chronic burn wounds, they take this bone marrow from the autologous bone marrow from the patient themselves and then flush it out onto the wounds, and that shows a lot of healing. Not only that, there has been a lot of work going on in my particular city in Hyderabad. It's the first in the world to initiate with the limbal transplantation of limbal stem cells, that is the stem cells of the eye, and that has shown to help in the corneal regeneration, and that is one of the example of it is happening. And of course, liver regeneration, we are technically in our lab, we are utilizing preclinical models, but then there had been a couple of clinical trials goes on into the in the clinic for liver regeneration. But to add you, in the present situation of COVID-19, MSC therapy had shown very good effectivity when being transplanted into the COVID-19 patients at a particular window period of time when they are being admitted in the ICU. At a very initial stage of the ICU, I must say that people will love to know that at a very initial stage of the ICU, when the patient enters, when these patients have been transplanted with the MSCs, allogenic MSCs, that they have shown very beautiful effect, and the patients have come out of the ICU and they have survived. So there is a hope, there is a point of future hope that is going on for MS stem cell therapy in our life.
Okay.
Any questions? Madhuchandra Durai.
Duari.
Stem cell therapy, how much effective? Okay, Duari, sorry. I apologize for mistakenly pronounced. Stem cell therapy, how much effective in autism treatment is? Very good question. Yes, indeed. In autism, there are quite a bit of therapeutical efficacies. I know my collaborators as well as others are doing it in clinics, trying to take it to the clinics in US, but definitely would be more ethical for them to answer on behalf of those particular study results. Yes. Yeah, that's very good. Zulfikar Mehdi. Wonderful. I guess I have answered your question. It is known that mesenchymal stem cells are naturally resistant to COVID-19 because of no ACE receptors. So why don't we hear about any possible therapeutic use? Indeed, Zulfikar, they are being tried. In fact, I was being approached by one of the company here, Therapeutic in India, who applied for clinical trials of MSCs. Yes, indeed, they are ACE receptor negatives, and you know how does it work? It basically works by the immune, because MSCs are immunomodulators. They secrete their immune modulations, and they help in secreting anti-inflammatories, and that actually negates out the inflammatory, the cytokine storm that happens in the COVID-19 patients at a very early phase. As I mentioned, if they have been given, they have showed positive effect. In India also it has been tried, but in outside India, in Europe, that has been tried a lot, and they have shown positive results. And there are a lot of clinical trials been happening world across. Yes, the next question, if I have time, can I take the next question?
Yes.
Yes, sir.
Okay, Acharya Arthur. Okay. Hi, sir. I'm a student. How can we address the risk of insertional mutagenesis and oncogene transactivation associated with the first generation of gamma retroviral vectors? A very, very pertinent question. Very good question. I must appreciate, Acharya, you have put forward a very beautiful question, and I appreciate your question because you have been thinking and you have been studying, you have been reading about it. So that's a fact of it. The lentiviral vector systems, what we have utilized, and we have shown efficacy in a preclinical model, that is true, that happens, that has shown an effect. But we do have at the back of our mind the doubt of insertional mutagenesis. And to avoid that, we in our lab, as well as others in the field, we know that we are trying to utilize CRISPR-Cas9 technology to avoid the insertional mutagenesis and to control it and utilize it to avoid that particular insertional mutagenesis. But definitely, that if we can avoid that, and the efficacy can be increased much higher. Yes, Dr. Jana, would you like to take them to the next questions? Do you have any questions? Oh, I appreciate that. You are a class 12 student, and I appreciate you. I see your interest onto it. I very much like your question, and I appreciate that. If you have any questions, feel free to write to me. I'll be happy to guide you through. Yes. Any other questions?
Amitabh, if you
Yes.
scroll up, then you can find more questions. I think you started from. Yes,
true, very true, Videsh. Yes,
there are more questions above
yes, after Yes, Srijani Haja asked about after transplantation, can the stem cell lost its regenerative property? It's a $1 million question. I'm sorry. Harith, I'll call you back. I'm addressing a webinar. I'll call you back. Sorry about that. The question was, I'm sorry, I lost it. Stem therapy. I'm sorry. Any other question? Which question I was in?
The question is from
What is the
Srijani
life?
Haja.
Okay, can you repeat the question, please? Okay, I got it. Stem cell therapy, how much effective in case of, no, that was not the question. There were other questions too.
There is, after transplantation, can the stem cell lost its regenerative property? This is the question from Srijani Hazra.
Oh, Srijani, if you have heard my lecture keenly, I told you that there were 3 challenges that we always have in the stem cell research. And the stem cells, right? If we transplant the stem cells has to proliferate and differentiate finally into a tissue-specific regeneration. So at one point of time, they have to lose their stemness and become a differentiated cells. If they remain as stem cells, they can only be secreting the trophic factors at the site of the injury, but cannot. The fate of the stem cell cannot be studied. To some extent, when there had been shown to be effective when MSCs have been transplanted, the transplanted MSCs have been shown to release these trophic factors that helps in the regeneration of the tissue, injured tissue. But of course, not only that, our school of thought is that, or my specifically the school of thought is that it not only has a trophic factor release, but also what it does is the fate of that stem cell can also be regenerated, which again we are trying to prove with multiple different to show that yes, the transplanted stem cells can regenerate into a tissue. For regeneration of the tissues, it has to leave behind its stemness and go in towards a differentiated phenotype. Okay, does it answer your question?
Thank you, sir, for your valuable input relating to these questions. And sir, due to the time limitation, we have just ended the question answer session, and I'm requesting Dr. Madhuchanda Banerjee to forward as prescribed the session.
Thank you, Sri Montaj Mondal. Thank you, Dr. Amitabh Dash. It was a real pleasure listening to you. It was a very informative and descriptive lecture, and it was very good. I mean, it was, very nicely represented, and we had great pleasure listening to that. We hope that we'll get you again to hear from you again later. So thank you so much once again.
I totally enjoyed presenting students. Yes. Please
thank you so much. Thank you so much. So we will now proceed with we have come to the final part of the session. I would now request Dr. Kajal Nayan Mojumdar to kindly give away the vote of thanks and end the program.
Thank you, Mojumdar. Good evening. Being the organizing secretary of the webinar, I am feeling privileged to deliver the vote of thanks on behalf of the organizing committee and Department of Zoology, Midnapore College. Firstly, I would like to express my immense gratitude to our respected principal, sir, Dr. Gopal Chandra Bera, for his constant encouragement and motivation in planning and holding this webinar. I would extend my sincere appreciation to the honorable speakers, Dr. Videsh Mahato from Department of Pathology, University of Cambridge, and Dr. Amitabh Dash, Principal Scientist from Department of Applied Biology, Indian Institute of Chemical Technology, for accepting our invitation and taking out time to deliver lectures, which are of great value to all the attendees, as well as the students who are thinking of doing research in this area. I also extend special thanks to all the faculty members of our department who have come together to make this event a success. Finally, a big thank you to all of you, all the attendees, without whom this webinar would not have been possible. Thank you. Thank you very much. So we are concluding here. Thank you. Thank you very much.
Partho. Is there any way to answer those questions? So can we discuss? So your microphone is muted, Partho.
Yes. I think that all the participants have moved. Particularly the questions was the question I was thinking of. It was from one of my student, Konisha Kaur. Is there any way out that I can send you the message via email or something, so that you can?
Fine. Yeah, that should be fine, and I can answer in the YouTube also. If I don't know what's the plan, but if
I'll send you the questions. I'll get the email from Gautamda and I'll send you the question. Actually,
okay,
that lady, that student, she is working on your field, I think. So she might have some questions. He will be privileged to communicate with you. So I'll get the question from her and let it know, get it to you by email.
Yeah, that would be great. I love questions actually.
Okay, thank you. Thank you. Thank you all. আমরা কি সেশনটা এখানে শেষ করব?
হ্যাঁ, কন্টিনিউ করতে পারলে